For nearly 70 years, scientists thought they understood this fish. They had specimens, they had diagrams, they had descriptions in textbooks. Every time one was brought up from the deep ocean, researchers examined it, recorded what they saw, and moved on. The fish was cataloged, understood, filed away. Every single one of those records was wrong because every specimen brought to the surface had already destroyed its own secret before anyone could examine it. As the fish rose through changing pressure, the structure that defined it, the thing that made it extraordinary, collapsed. What arrived in the net looked nothing like what had been hovering in the darkness below, and for nearly 70 years, no one knew the difference. The Barreleye fish had been hiding the truth by dying before humans could see it alive. When researchers finally filmed one intact in 2004, what the footage showed stopped people cold. The head was covered by a fully transparent dome, a fluid-filled shield, completely see-through, that had never appeared in any drawing or description. Inside it, two vivid green eyes were rotating slowly within the dome, tracking movement, pointing in directions no eye on any known fish can point. And the two dark spots on the front of the fish that every diagram had labeled as eyes. Nostrils. Not one researcher over nearly 7 decades had been looking at the eyes. They had been looking at the nose. The entire fish had been misread, from the outside in, because it had never once been seen the way it actually exists. And the deeper you look at what this fish actually is, how it sees, how it hunts, how it survives in one of the most lightless environments on Earth, the stranger it gets. But before we continue, let me know in the comments if you have any topic suggestions you would like me to cover in future videos. Now, let's descend into the Twilight Zone and look at the fish that forced science to rethink how vision itself can work in the deep ocean. Far beneath the restless skin of the Pacific, below the paths of whales and the last silver flashes of schooling fish, the ocean begins to lose the sun. The fading happens slowly at first. Blue light thins into dim cobalt, then into something softer and stranger, as though the sea itself is forgetting daytime. Between roughly 200 and 1,000 meters below the surface lies the mesopelagic zone, a region sailors once imagined as empty simply because human eyes could not follow the light that far. Oceanographers later gave it another name, the Twilight Zone, not because it is fully dark, but because daylight arrives there, weakened and incomplete, dissolving long before it can touch the seafloor far below. At 200 meters, barely 1% of the surface sunlight remains. The water still carries a faint memory of morning, enough for silhouettes to drift across the dim blue ceiling overhead. By 600 meters, even that memory has almost vanished. The ocean becomes an enormous volume of near blackness, where shapes exist before colors, and movement matters more than detail. Down here, the difference between seeing and not seeing may be measured by a single particle of light slipping through the water from far above. Nothing grows in this place. No forests of kelp sway through the currents. No fields of algae stretch toward the sun. Photosynthesis ends long before the Barreleye's world begins. The Twilight Zone survives on fragments descending from brighter waters overhead, a constant slow snowfall known as marine snow, dead plankton, drifting fecal pellets, scraps of tissue, shattered scales, microscopic corpses. All of it falls through the water column in a silent downward migration that never truly stops. The deep ocean feeds on what the surface forgets. It is an immense habitat, larger by volume than any environment on Earth, stretching across nearly every ocean basin on the planet. Yet despite its scale, it remains among the least explored regions humanity has ever encountered. Fewer human beings have visited these depths than have walked across the surface of the moon. Entire populations of living animals move through the darkness unseen, carrying out migrations, hunts, and reproductive cycles that science still understands only in fragments. Somewhere between 400 and 800 meters below the surface, suspended in the center of this dim and pressurized world, the Barreleye drifts almost motionless. Every detail of its body belongs specifically to this environment. The cold remains nearly constant, usually between 2 and 4 degrees Celsius, a temperature stable enough to slow metabolism and conserve precious energy. Food arrives unpredictably. Movement becomes expensive. Survival favors patience over speed. The pressure shapes everything most of all. At these depths, the ocean presses inward with roughly 40 to 80 times the atmospheric pressure experienced at sea level. Human lungs exposed directly to such conditions would collapse almost instantly. Air spaces become liabilities in the deep sea, crushed by the immense weight of water overhead. Flesh, organs, and skeletons must adapt or fail. The Barreleye's strange anatomy did not emerge from creativity or chance. It emerged from millions of years spent negotiating the exact physical demands of this narrow band of darkness. Even the water behaves differently here. Currents slow into long, invisible rivers. Sound travels farther than light. Small disturbances linger. A drifting crustacean may appear suddenly out of the blackness, like a falling star, then disappear again into the cold. In this suspended world between surface and abyss, life becomes quiet, economical, and extraordinarily precise. The Twilight Zone is often imagined as empty space between more familiar realms, neither the bright ocean above nor the abyss below. Yet for the Barreleye and countless other animals hidden in the mesopelagic dark, this narrow band of fading light is not a passage between worlds. It is home, ancient and complete, shaped by pressure, hunger, and the last weak traces of the distant sun. In museum drawers and glass jars scattered across marine laboratories, the Barreleye spent decades existing in a form that was not entirely real. Preserved in cloudy alcohol, pale from decompression, and torn by nets hauled upward through hundreds of meters of water, the fish appeared small, damaged, and strangely ordinary compared to the living animals suspended far below the surface. Scientists gave it a name in 1939, Macropinna microstoma, and from that moment onward, its anatomy entered textbooks, field guides, and scientific records as something understood. Yet almost every detail that made the animal extraordinary had already vanished before anyone examined it. The journey to the surface was the problem. Deep-sea trawl nets do not lift animals gently. They scrape through darkness like moving walls, gathering whatever cannot escape quickly enough. Pressure changes begin immediately during ascent. Soft tissues swell, distort, and rupture. Delicate gelatinous structures collapse under conditions they were never built to survive. By the time a Barreleye reached the deck of a research vessel, the fish no longer resembled the living creature drifting through the mesopelagic dark only hours before. Researchers examining those early specimens consistently described 2 dark circles near the front of the head. They appeared symmetrical, glossy, and positioned where fish eyes would normally sit. Scientific illustrations reproduced them carefully, often exaggerating their size to emphasize what seemed like unusually large forward-facing eyes. The assumption settled quietly into accepted fact, repeated from paper to paper over the course of decades. The Barreleye became known as another strange deep-sea fish with oversized vision adapted for darkness. What nobody realized was that the eyes were somewhere else entirely. The transparent dome that enclosed the top of the fish's head, a soft fluid-filled shield, unlike anything seen in most vertebrates, could not survive decompression. Removed from the immense pressure of deep water, it collapsed almost immediately, folding inward into shapeless tissue before the specimen ever reached scientific observation. Without the dome intact, the actual anatomy of the fish became almost impossible to interpret. Hidden behind the collapsed membrane sat the true eyes, large green tubular organs pointed upward toward the faint traces of distant light. They remained concealed inside the damaged head, while the dark spots on the front, not eyes at all, took their place in the scientific imagination. Those spots were olfactory organs, structures used for detecting chemical traces drifting through the water column. They functioned more like nostrils than vision, quietly sampling the ocean for invisible gradients of scent carried by the currents. For nearly 70 years, scientists unknowingly mistook the fish's sense of smell for its gaze. The scale of the misunderstanding spread far beyond a mislabeled diagram. Anatomical descriptions, museum displays, educational illustrations, all of them reflected an animal that did not truly exist in nature. The Barreleye became a scientific reconstruction assembled from a body already altered by death and pressure loss. Every preserved specimen carried the same silent distortion. The most extraordinary feature of the fish had been hidden inside the exact structure least capable of surviving the trip into human hands. There is something strangely fitting about the way the error persisted. The deep ocean does not surrender information easily. Its creatures are shaped by conditions so different from the surface world that the act of retrieving them can erase the very details researchers hope to study. In the case of the Barreleye, the evidence destroyed itself with remarkable consistency. Generation after generation of scientists looked directly at the fish while missing the living geometry concealed inside its transparent dome. Far below the surface, meanwhile, the real animal continued drifting through the cold water unchanged, carrying its upward-facing eyes inside a fragile chamber of fluid and light, untouched by the diagrams that tried for decades to define it. Along the coast of central California, where cold Pacific currents fold against the continental shelf, and underwater canyons descend into darkness, only a short distance from shore, a group of marine researchers spent years lowering machines into parts of the ocean human beings could never physically reach. The Monterey Bay Aquarium Research Institute, known more quietly by its initials, MBARI, became one of the few places on Earth where the deep sea could be observed without immediately destroying what lived there. Instead of dragging animals upward through nets and pressure shifts, the Institute sent cameras downward into the water itself, allowing fragile organisms to remain inside the conditions that shaped them. Their primary tools were remotely operated vehicles, small unmanned submarines connected to a research vessel above by long tether cables, carrying electricity, data, and control signals through the water column. Inside dark control rooms on the surface, pilots guided these machines slowly through the mesopelagic depths, while high-definition cameras transmitted live images upward in real time. Floodlights cut pale tunnels through the darkness ahead of the vehicle. Mechanical arms drifted folded at the sides like sleeping limbs. The ocean, for the first time, could be watched without being immediately broken apart by capture. In 2004, during one of these dives, the cameras encountered a living barreleye. The contradiction appeared almost immediately. Researchers recognized the fish by its small body and hovering posture, yet the head looked entirely wrong compared to the accepted scientific illustrations. Instead of a flattened face with visible forward eyes, there was a smooth, transparent dome arching over the front of the skull, clear enough for the internal anatomy to remain visible through the tissue itself. Suspended inside the dome were two luminous green cylinders pointing upward into the water above, perfectly aligned toward the dimmest traces of downwelling light. The eyes were not where science believed they should be. The footage revealed the animal hovering with extraordinary stillness, barely moving except for delicate adjustments of its fins. The dome caught the ROV lights and bent them softly through the fluid-filled shield, producing faint reflections across its curved surface. Inside, the green eyes glowed with an almost manufactured precision, like instruments mounted behind polished glass. Nothing about the fish resembled the damaged specimens preserved in museum collections. The living Barreleye looked less like a reconstructed deep-sea curiosity and more like an organism designed around optical physics itself. As the vehicle maintained position nearby, the cameras continued recording details no scientist had ever directly observed before. The transparent shield remained perfectly intact under pressure. The eyes sat fully enclosed inside the dome, protected within their chamber of fluid. Even the shape of the head altered previous assumptions about the species. Decades of accepted anatomical descriptions quietly unraveled in front of the monitor screens aboard the research vessel. When the observations were formally published in 2009 in the journal Copia, researchers included a complete redescription of the species based on the living footage. The scientific record itself required correction. Diagrams changed, textbooks updated. Features that had been overlooked for nearly 70 years suddenly became central to understanding the fish at all. Outside scientific circles, the video spread widely across the emerging internet, accumulating millions of views and becoming one of the most recognized pieces of deep-sea footage ever recorded. Audiences accustomed to imagining the deep ocean through damaged specimens or artistic reconstructions were suddenly looking directly at a living animal inside its own environment, moving slowly through the dark exactly as evolution had shaped it to move. Yet the significance of the moment reached beyond the Barreleye itself. The footage exposed a larger problem, resting quietly beneath decades of marine biology. Much of humanity's understanding of deep-sea life had been built from bodies altered by decompression, fragile tissues destroyed during collection, behaviors erased before observation ever began. The Barreleye was not simply a misunderstood species. It became evidence that entire categories of deep ocean anatomy may have been distorted by the very methods used to study them, hidden truths collapsing on their journey toward the surface world. Most vertebrate animals carry their skulls like sealed walls, dense barriers of bone separating delicate sensory organs from the surrounding world. The Barreleye followed a different path. Over millions of years in the mesopelagic dark, the front of its head transformed into something closer to a window than a shield, a transparent dome arching over the eyes with such clarity that the internal structures remain visible through the tissue itself. Among known vertebrates, almost nothing comparable exists. Other deep-sea fish evolved large eyes, reflective organs, or bioluminescent features, but the Barreleye alone enclosed its visual system inside a fluid-filled chamber suspended beneath a transparent surface. The dome is not glass-like in the rigid sense suggested by photographs. It is soft, flexible tissue, technically derived from modified scale structures, and filled with a clear fluid whose optical properties closely resemble the seawater surrounding the fish. At depth, under immense external pressure, the structure maintains its smooth curved shape with remarkable stability. Removed from that environment, however, the dome loses integrity rapidly. Without the compression of hundreds of meters of water holding the tissues in balance, the membrane folds inward and collapses within minutes, becoming almost unrecognizable by the time preserved specimens reach the surface. Its existence appears tied completely to the physics of one narrow region of the ocean. The Barreleye's dome is not an anatomical curiosity that could function broadly across different habitats. It belongs specifically to the Twilight Zone, calibrated for a world of near darkness, cold water, and crushing pressure. Even slight changes in those surrounding conditions alter the structure itself. Researchers studying the dome believe its primary role is optical. At depths between roughly 600 and 800 meters, sunlight no longer illuminates the water in any meaningful sense. Instead, faint residual photons drift downward in scattered traces, thin enough that most animals would perceive almost nothing at all. The curved surface of the dome may help gather and channel that scarce light toward the fish's tubular eyes, functioning as an additional optical layer above the already specialized visual system hidden beneath it. Rather than allowing light to disperse randomly across the head, the dome appears to preserve and direct whatever illumination still exists in the water overhead. The fluid inside contributes to this effect. Because its refractive index remains extremely close to that of seawater, light passes through the dome with minimal distortion. The boundary between ocean and tissue becomes optically subtle, reducing scattering that might blur incoming images. In practical terms, the fish carries its visual chamber through the water without significantly disrupting the fragile light patterns it depends upon to survive. Protection may be equally important. The Barreleye's eyes rank among the most sensitive photoreceptive organs known in vertebrate animals, specialized for detecting dim contrasts that would be invisible to most forms of life. Such delicate structures require shielding from physical damage in an environment filled with drifting organisms, suspended particles, and the occasional stinging tentacles of gelatinous predators. The dome forms a barrier between the eyes and the surrounding water, while preserving visibility, protecting the organs without enclosing them behind opaque tissue. There are hints that the structure may also shape the movement of water itself. As the fish hovers and subtly rotates in place, the smooth curve of the dome likely reduces turbulence across the head, allowing water to flow around the animal with minimal disturbance. In a habitat where conserving energy matters constantly, even slight improvements in hydrodynamic efficiency become meaningful over evolutionary time. The dome ultimately represents a solution so precise that it feels inseparable from the environment that created it. Every feature depends upon exact conditions: pressure strong enough to stabilize the tissue, temperatures cold enough to preserve metabolic balance, and darkness deep enough for extraordinary vision to matter at all. The structure does not simply survive in the Twilight Zone, it only makes sense there, as though the surrounding ocean shaped a transparent chamber specifically for the thin remaining light of the deep Pacific water. In the deep ocean, vision follows different rules than it does near the surface. Sunlight no longer floods the environment evenly, scattering across reefs or reflecting from open water. Instead, illumination arrives sparsely, reduced to faint traces descending from far above. Animals living in this dim world cannot rely on broad panoramic sight. Evolution narrows their focus. It concentrates sensitivity into precise directions where the remaining light still exists, shaping eyes less for beauty than for extraction, gathering every available photon before it disappears into darkness. The Barreleye's eyes belong to this narrow and highly specialized tradition. Rather than rounded spheres positioned for wide vision, each eye forms a long green cylinder enclosed within the transparent chamber above the fish's face. This structure is known as a tubular eye, a design that has appeared independently in several deep-sea lineages over millions of years. Different animals arrived at similar solutions because the optical problem remained the same. In the mesopelagic zone, there is almost no light to waste. Tubular eyes function less like ordinary fish eyes and more like highly compressed telescopes. Instead of collecting visual information from all directions equally, they focus intensely on a restricted cone of water directly overhead. The trade-off is severe but effective. Peripheral awareness becomes limited, yet sensitivity increases dramatically. In practical terms, the fish sacrifices breadth of vision for precision in a single upward direction, exactly where faint downwelling light continues filtering through the ocean from the distant surface. Inside each tubular eye rests a dense concentration of rod cells, the photoreceptors specialized for low-light environments. Human eyes also contain rods, primarily used during nighttime vision. But the Barreleye's visual system pushes this adaptation far beyond surface standards. Cone cells, which process color and perform best under bright conditions, are almost entirely absent. The fish perceives its world largely in shades of brightness and shadow, an existence built around contrast rather than color. In the dim pressure-filled water where it lives, distinguishing the silhouette of prey against weak light matters far more than perceiving vibrant detail. The lenses themselves are unusually large relative to the fish's body size. This enlargement increases the amount of incoming light entering the visual chamber, allowing the eyes to collect faint signals that would otherwise vanish unnoticed into the surrounding dark. Every anatomical proportion reflects the same underlying pressure from evolution: preserve sensitivity at all costs. Even the strange green appearance of the eyes serves a functional purpose. The coloration does not originate from the surrounding fluid or the transparent dome, but from a yellow pigment filter embedded within the lenses themselves. Among deep-sea fish, colored lenses remain exceptionally rare. Researchers believe the pigment helps the barreleye separate useful, visual information from the overwhelming blue glow common throughout mesopelagic waters. Many organisms at these depths produce blue bioluminescence, brief flashes and drifting pulses that scatter through the water like distant electrical signals. The yellow filter appears to reduce some of that optical noise, sharpening the contrast between prey and background illumination. This tuning gives the barreleye a highly specific visual target. Its eyes are calibrated not for scanning the darkness broadly, but for watching the water above, where tiny crustaceans, drifting gelatinous animals, and fragments of marine life pass between the fish and the last weak traces of descending light. Against that dim ceiling, even transparent organisms reveal themselves as moving silhouettes. The result is one of the most specialized vision systems known in the ocean, a pair of organs refined for a single task in a single direction. Yet, embedded within this adaptation is a quiet contradiction. The fish hunts objects drifting overhead, but feeding requires movement forward through the water. The eyes remain pointed upward, while the mouth waits below, creating a strange geometric problem within the animal's own anatomy. For years, researchers struggled to understand how such a visual arrangement could function during actual predation. How a fish could pursue food it seemed incapable of directly facing. For decades, marine biologists, looking at preserved barreleye specimens, carried a quiet uncertainty beneath the accepted diagrams. Even after the true position of the eyes became known, another difficulty remained unresolved. The visual system seemed almost impossibly specialized for watching the water above, yet the fish itself was still a predator, dependent on accurately approaching drifting prey through darkness. The geometry appeared wrong. A hunter cannot survive long if its gaze remains permanently directed away from the direction of attack. The answer revealed itself slowly, through the living footage gathered during deep-sea observation. Inside the transparent dome, the green tubular eyes were not fixed structures anchored permanently toward the surface. They moved. Researchers watching the Barreleye at depth observed the eyes pivoting within the fluid-filled chamber itself, rotating from their upward position into a forward-facing orientation while remaining completely enclosed inside the dome. The motion appeared smooth and deliberate, almost mechanical in its precision. One moment the cylinders pointed vertically through the roof of water overhead, tuned toward faint silhouettes, drifting against the dim remains of descending light. Then, with subtle muscular adjustment, they shifted forward through the transparent shield, aligning directly with objects ahead of the fish. The contradiction dissolved immediately. The Barreleye had never been trapped within a single direction of sight. It possessed two visual modes contained within the same anatomical system. In its upward position, the eyes function as detectors, scanning the weakly illuminated water above for passing shadows. Small crustaceans, gelatinous organisms, and drifting fragments crossing between the fish and the distant surface become briefly visible as interruptions in the faint overhead glow. The Barreleye hovers beneath them with extraordinary patience, conserving movement while its visual system performs the difficult work of detection. Once prey enters striking range, the eyes rotate forward. The fish no longer observes distant silhouettes, but directly tracks the target ahead through the dome itself. The shift transforms the visual system from passive surveillance into active targeting. Few animals possess such a dramatic transition between modes of perception inside a single pair of eyes. Among vertebrates, nothing else quite matches this arrangement. Tubular eyes already represent an extreme adaptation for dim environments, sacrificing broad awareness for concentrated sensitivity. Yet, other fish possessing tubular eyes generally remain constrained by their fixed orientation. The Barreleye alone redirects these elongated visual organs through a transparent external chamber while preserving optical clarity. Evolution produced not only a specialized eye, but a movable one enclosed within its own sealed observatory. The mechanics behind the motion reveal further specialization hidden beneath the dome. The muscles controlling the eyes are unusually large relative to the fish's body size, capable of carefully rotating the cylinders within their sockets while maintaining stable alignment. In most fish, eye movements remain comparatively simple, small directional adjustments coordinating with rapid swimming behavior. The Barreleye depends on something more precise. Its survival requires the ability to shift between vertical detection and forward pursuit without disrupting the fragile visual information gathered from the surrounding water. Everything about the movement reflects the slow economy of life in the mesopelagic zone. The fish does not chase widely or waste energy on rapid searching. Instead, it waits suspended in the cold darkness, allowing the ocean itself to carry potential prey overhead. A drifting jellyfish, a small copepod, or a suspended crustacean crossing the faint field of downwelling light becomes visible first as contrast rather than detail. The eyes detect the movement above, rotate forward, and guide the fish into an exact approach. In this sense, the Barreleye's eyes are not merely sensory organs in the ordinary biological meaning. They function as the animal's primary hunting apparatus, integrating surveillance, targeting, and spatial orientation into a single rotating system hidden behind transparent tissue. The fish survives not through speed, strength, or aggression, but through the careful redirection of sight itself, adjusting its gaze inside the dome with quiet precision while the surrounding ocean drifts endlessly past in darkness. Movement in the mesopelagic zone carries a cost. Every burst of motion burns energy that may not be replaced for hours or even days, and every disturbance in the water risks revealing a predator's position to other unseen animals drifting nearby. The barreleye survives by avoiding unnecessary motion almost entirely. Researchers observing living specimens often describe them suspended in the water column with such steadiness that they appear less like swimming fish and more like objects carefully hung in darkness, maintaining position through tiny adjustments, too subtle to notice at first glance. This stillness is not inactivity. It is a hunting method shaped slowly through evolutionary time, refined under conditions where patience yields more success than pursuit. The barreleye does not roam widely searching for prey. Instead, it positions itself beneath drifting colonial organisms and waits. Among the most important of these organisms are siphonophores, fragile predators assembled not as single animals, but as floating colonies, composed of many specialized bodies working together. Some siphonophores extend through the water in long, transparent chains, stretching for tens of meters, their trailing tentacles suspended almost invisibly in the dark. Along those tentacles hang thousands of microscopic, stinging structures called nematocysts, each capable of firing tiny, venomous harpoons into passing prey. Small crustaceans and drifting zooplankton become trapped almost immediately upon contact, immobilized before being transported upward through the colony for digestion. The barreleye has learned to exploit this system without becoming prey itself. Direct observations from deep-sea footage revealed the fish approaching siphonophore tentacles carefully, maneuvering among the suspended feeding structures while targeting the small captured organisms trapped along them. Rather than hunting the prey directly through open pursuit, the barreleye steals food already collected by another predator. This behavior, known as kleptoparasitism, allows the fish to conserve energy while taking advantage of the siphonophore's immense filtering network drifting silently through the mesopelagic water. The transparent dome above the barreleye's head appears central to making this strategy possible. While originally evolved as part of the fish's visual system, the dome likely provides physical protection as well, shielding the sensitive eyes from accidental contact with stinging tentacles during feeding. Most fish approaching siphonophore structures risk injury to exposed tissues, particularly around the face and eyes where nematocysts can discharge instantly. The barreleye carries a smooth fluid barrier over its visual organs, allowing it to navigate among the tentacles with a degree of protection unavailable to many other predators at similar depths. The result is an unusual overlap between sensory adaptation and feeding behavior. A structure evolved primarily for gathering light in darkness also enabled access to a food source difficult for competing animals to exploit safely. Evolution often builds new strategies from existing anatomy rather than creating entirely separate systems, and the barreleye embodies this layered efficiency with remarkable precision. The actual hunting scene unfolds with extraordinary quietness. A siphonophore drifts through the water like a transparent chain suspended in empty space, almost invisible except where faint biological light catches along its body. Around it hangs a curtain of tentacles, extending downward through the cold water. Beneath the colony, nearly indistinguishable from the darkness itself, the barreleye waits motionless. Its body absorbs what little light remains in the environment, while the transparent dome reveals almost nothing except the faint green cylinders of the eyes, turning slowly upward. Tiny prey animals become visible first, as interruptions in the dim overhead field, then as trapped bodies trembling along the siphonophore's feeding lines. The fish rises carefully among the tentacles, rotating its gaze forward as it approaches, There is no sudden chase, no violent strike erupting from the dark. Only controlled movement through suspended danger, the patient theft of food from another hunter's trap, and then a gradual return to stillness beneath the drifting colony once more. In the deep ocean, survival often belongs not to the fastest predator, but to the one that spends the least energy becoming visible at all. Darkness in the mesopelagic ocean is never completely empty. Even far below the reach of meaningful sunlight, the water flickers constantly with brief signals produced by living bodies. Tiny blue pulses drift through the currents like distant sparks. Soft flashes appear and vanish without warning. Thin chains of light ripple downward through the blackness before dissolving again into invisibility. To human eyes descending suddenly into this environment, the deep sea might resemble a night sky turned upside down, filled not with stars but with organisms carrying their own illumination through the water. This phenomenon, bioluminescence, is among the most widespread forms of communication in the Twilight Zone. Roughly 76% of animals living in the mesopelagic depths are believed to generate light biologically, using chemical reactions inside specialized tissues to produce controlled flashes, glows, or pulses. In a habitat where sunlight fades almost completely, life began manufacturing its own visibility. The purposes vary across species, though certain patterns emerge repeatedly throughout the deep ocean. Some animals use light as a lure, attracting smaller organisms close enough to capture. Others flash rhythmic signals to locate mates drifting through the enormous darkness. Many species employ a subtler strategy known as counter-illumination, producing faint light along the underside of the body that matches the dim residual glow descending from above. Seen from beneath, the animal's silhouette disappears into the background light, erasing the shadow that predators would otherwise detect. For the Barreleye, this illuminated world creates both opportunity and danger simultaneously. Bioluminescent flashes reveal movement, outline drifting organisms, and expose the presence of nearby life. At the same time, every pulse of light may belong to another predator navigating the same water. Visibility in the mesopelagic zone rarely arrives without risk attached to it. To notice another animal often means becoming noticeable in return. The yellow pigment filter, embedded within the Barreleye's lenses, appears deeply connected to this environment of constant blue glow. Most mesopelagic bioluminescence falls within blue wavelengths because blue light travels farther through seawater than most other colors. Over evolutionary time, the deep ocean became saturated with blue optical signals layered continuously across the darkness. Researchers believe the Barreleye's yellow filter may function as a kind of visual refinement system, reducing portions of the surrounding blue haze and sharpening the contrast of specific shapes moving against it. Instead of being overwhelmed by countless, scattered flashes, the fish isolates meaningful silhouettes hidden within the luminous background. Around the Barreleye drift other animals equally specialized for this world of living light. Hatchetfish move through the water with flattened silver bodies lined beneath by rows of tiny photophores, light-producing organs arranged carefully along the belly. These organs emit controlled blue illumination, matching the weak downwelling light from overhead, rendering the fish nearly invisible when viewed from below. Their bodies become optical illusions suspended in open water, shadows erased through precise biological mimicry. Nearby, firefly squid pulse with sudden electric blue displays that travel along their arms and bodies in shifting patterns. In shallower coastal waters near Japan, entire swarms occasionally illuminate the ocean surface in vast glowing blooms. though individuals also descend into deeper mesopelagic layers, where their flashes punctuate the darkness below. Among the most specialized predators in this environment is the dragonfish, long-bodied and dark as the surrounding water itself. Some species possess the remarkable ability to generate red bioluminescent light, an adaptation almost invisible to most deep-sea animals whose eyes cannot detect red wavelengths at all. The dragonfish effectively carries a hidden searchlight through the ocean, illuminating prey while remaining unseen by the creatures it hunts. Within this crowded field of biological signals, the Barreleye navigates not by brute visibility, but by interpretation. Its rotating eyes and filtered lenses allow it to read subtle contrasts, drifting among flashes, shadows crossing faint glows, prey suspended briefly against illuminated water before vanishing once more. The mesopelagic zone becomes less a place of darkness than a shifting language of light, spoken continuously by the organisms inhabiting it. The Barreleye survives by learning that language with extraordinary precision, reading the deep ocean not as emptiness, but as an intricate field of moving signals suspended inside the cold Pacific dark. The transparent dome and rotating eyes draw attention so completely that the rest of the Barreleye often fades into the background, as though the fish ends at the edge of its own skull. Scientific illustrations linger around the strange architecture of the head, while the body behind it becomes little more than a dark extension trailing into obscurity. Yet, the deeper researchers looked at living specimens, the clearer it became that the animal's entire form is shaped with the same precision as its remarkable vision, every surface calibrated for a narrow existence suspended within the mesopelagic water column. The Barreleye itself is surprisingly small. Most known specimens measure between 6 and 15 centimeters in total length, roughly the size of a human hand. In photographs lacking scale, the fish can appear much larger because its anatomy feels so unusual, but in reality, it is a delicate animal drifting among much larger predators and colonial organisms in the deep Pacific. Its survival depends less on physical dominance than on invisibility, precision, and energy conservation. Viewed from the side, the body appears flattened laterally, compressed into a narrow profile that reduces resistance while hovering in still water. Unlike fast pelagic fish built for continuous swimming, the Barreleye does not carry a torpedo-shaped body optimized for speed. Its form resembles something quieter, more balanced than forceful, designed not for long pursuit but for remaining suspended with minimal effort. The fins reveal this immediately. Large pectoral fins extend horizontally from the body like narrow wings, constantly making tiny controlled adjustments, almost too subtle to notice. These fins provide stability and fine movement rather than thrust. The fish uses them to maintain exact positioning within the water column, drifting beneath prey fields or suspended tentacles without unnecessary displacement. In the mesopelagic zone, where food arrives unpredictably and calories remain scarce, avoiding wasted movement becomes a matter of survival. The Barreleye spends energy carefully, correcting its position through delicate fin motions instead of continuous swimming. Its body coloration contributes to the same strategy. The scales are small, dark, and highly absorbent to the faint light drifting through the environment. Rather than reflecting illumination outward, the body swallows it, dissolving against the surrounding water until the fish becomes difficult to distinguish from the darkness itself. Only the transparent dome interrupts this concealment, and even that structure remains optically subtle under natural conditions. The result is an animal that almost disappears, except for the specialized organs necessary to perceive the world around it. Near the front of the body sits a surprisingly small mouth. There are no oversized jaws or visible predatory adaptations associated with aggressive hunters of the deep sea. The opening remains narrow because the barreleye does not consume large prey. Its diet consists primarily of tiny crustaceans, gelatinous organisms, and suspended zooplankton gathered carefully from the surrounding water. The fish evolved not for overpowering prey, but for locating delicate drifting food in an environment where opportunities arrive slowly and unpredictably. Inside the body, another unusual detail appears in certain examined specimens. Many fish rely on a gas-filled organ known as the swim bladder to regulate buoyancy, adjusting internal pressure to remain suspended at specific depths with minimal effort. In the barreleye, this structure is greatly reduced or absent entirely in some individuals. Gas-filled organs become increasingly difficult to manage under the immense compression of deep water, and maintaining them can create complications during vertical movement. Instead, the barreleye appears to control its position actively through fin movement and body balance rather than passive flotation. Everything about the animal communicates specialization. The flattened shape, the stabilizing fins, the absorbent scales, the reduced mouth, the absence of obvious buoyancy structures, all of it reflects a creature refined for one precise layer of the ocean. The barreleye is not a generalist capable of adapting broadly across habitats. Its body belongs specifically to cold mesopelagic water, to drifting prey suspended in darkness, to slow hovering beneath distant traces of light. Even away from the famous dome above its head, the fish carries the unmistakable geometry of a life built entirely around one specific world. Most encounters between predator and prey in the mesopelagic zone happen invisibly. A drifting crustacean vanishes into darkness without sound or visible struggle. A gelatinous body disappears between faint currents. Small larvae suspended in open water simply cease moving as another animal intercepts them somewhere beyond the reach of observation. The barreleye participates in this hidden exchange quietly, feeding on some of the smallest and most delicate organisms drifting through the deep Pacific. Stomach content analyses from the limited number of collected specimens provide only fragments of the full picture, yet certain prey items appear consistently. Tiny copepods, among the most abundant crustaceans in the ocean, form part of the diet. Amphipods, small laterally compressed scavengers and hunters drifting through the water column, have also been identified. Hydromedusae, delicate jellyfish related to siphonophores, appear frequently alongside siphonophore zooids themselves, individual feeding bodies stolen from the colonies suspended above. Small fish larvae occasionally appear as well, soft-bodied juveniles carried downward from shallower waters into the mesopelagic depths. The pattern suggests opportunism within strict environmental limits. The barreleye does not seem to undertake large vertical migrations, searching for food across vast layers of ocean. Instead, it remains largely confined to its narrow depth range, feeding on whatever suitable organisms drift into reach. In the Twilight zone, movement itself becomes expensive. Rather than chasing distant prey, the fish allows the surrounding ecosystem to deliver food gradually through the water column, conserving energy whenever possible. This same environment provides partial protection from predators. The barreleye rarely appears in stomach analyses from larger animals, making direct confirmation of its predators difficult. Its small size, near invisibility, and remote habitat all contribute to the scarcity of evidence. Yet some likely threats emerge from what is known about mesopelagic food webs. Larger predatory fish moving through the same depths almost certainly consume barreleyes when opportunities arise. Among the strongest candidates is the lancetfish, Alopesaurus ferox, a long-bodied predator with oversized jaws and a reputation for consuming almost anything encountered in open water. Lancetfish range across mesopelagic and deeper pelagic zones, feeding opportunistically on smaller fish, squid, and drifting animals. Certain shark species, descending periodically into the twilight zone, may also prey upon barreleyes, particularly those adapted for hunting in dim environments where bioluminescent cues and subtle motion reveal concealed organisms. Against these threats, the barreleye relies primarily on avoidance rather than defense. It possesses no venom, no armored scales, no burst swimming ability capable of outrunning large predators. Instead, the fish minimizes the chance of detection altogether. The transparent dome blends optically into the surrounding water. The dark body absorbs faint illumination rather than reflecting it outward. The hovering behavior eliminates sudden motion that might attract attention from nearby hunters. In many situations, survival depends simply on remaining unnoticed long enough for danger to drift elsewhere. The economics of life at these depths reinforce this strategy continuously. Food remains scarce compared to surface ecosystems rich with sunlight and photosynthesis. Temperatures hover only a few degrees above freezing, slowing metabolic processes across much of the mesopelagic fauna. The barreleye likely feeds infrequently by surface standards, digesting slowly and conserving energy between successful captures. Rapid growth, constant movement, and frequent feeding would be impossible to sustain in such a nutrient-limited world. Yet despite the scarcity experienced by individual animals, the twilight zone collectively processes enormous quantities of biological material. Estimates suggest mesopelagic fish communities consume roughly 10% of the ocean's surface primary production, intercepting organic matter descending from above before it can sink deeper into the abyss. This represents a staggering transfer of energy, occurring largely out of sight, carried out by countless small predators suspended throughout the dark water column. The barreleye occupies only a tiny part of that hidden system, feeding quietly on drifting organisms while remaining prey itself to larger unseen hunters. Around it, the mesopelagic ecosystem continues operating with extraordinary scale and subtlety, vast amounts of life sustained by dim traces of productivity arriving slowly from the bright world far above. Each evening, as sunlight fades across the surface of the ocean, an immense movement begins beneath the water. Countless small animals rise upward from the mesopelagic depths toward the shallower layers above, ascending through the dark in numbers so vast that the migration outweighs every herd movement on land combined. Copepods, shrimp-like crustaceans, lanternfish, gelatinous drifters, and microscopic plankton all participate in this slow nightly ascent, feeding closer to the surface under the protection of darkness before descending again at dawn. Scientists refer to the phenomenon as vertical migration, a rhythmic movement repeated across nearly every ocean on Earth for millions of years. The scale is difficult to fully imagine because almost all of it happens unseen. Entire populations shift hundreds of meters through the water column each day, forming a living tide moving silently between light and darkness. Sonar systems aimed into the twilight zone often detect dense scattering layers, rising and falling through the ocean at sunset and sunrise, not geological structures or currents, but enormous communities of animals migrating together through open water. The barreleye appears to occupy a different role within this cycle. Unlike many mesopelagic fish that travel long vertical distances each night, it is generally considered a resident species, remaining within a comparatively stable depth range instead of ascending fully toward surface waters. Its anatomy reflects this relative permanence. The hovering behavior, the specialized upward vision, the delicate energy economy, all suggest an animal adapted for remaining suspended within the deeper twilight zone rather than commuting between ecological layers every day. Yet, even a resident predator cannot remain entirely disconnected from migration when its prey participates in it continuously. Many of the small organisms consumed by the Barreleye, particularly copepods and drifting crustaceans, move upward each night before returning downward again with the coming daylight. The fish's food supply effectively travels through the water column twice every day, carrying energy from surface ecosystems into deeper regions of the ocean. Researchers suspect the Barreleye may respond by making smaller vertical adjustments of its own, perhaps shifting upward or downward by 100 to 200 meters to remain aligned with concentrations of migrating prey, while avoiding the energetic cost and predation risks associated with full migration. Such movements would fit the fish's broader strategy of conservation and precision. Rather than participating in the massive nightly ascent directly, the Barreleye appears to position itself carefully within the moving flow of organisms, feeding selectively as prey passes through its preferred depth range. The ocean itself delivers food downward and upward in cycles, and the fish remains suspended within that circulation, almost like a stationary observer inside a moving river. This migration carries consequences extending far beyond individual predators and prey. Every night, organisms feeding near the surface consume carbon-rich material generated through photosynthesis in sunlit waters. When those animals descend again into deeper layers, they transport that carbon downward inside their bodies, waste products, and eventual remains. Scientists call this process the biological carbon pump, one of the most important systems regulating the chemistry of Earth's atmosphere and oceans. Collectively, diel vertical migration is estimated to move roughly 10 billion tons of carbon toward the deep ocean each day. Without this continual transport, far more carbon dioxide would remain near the surface atmosphere rather than being carried into deeper marine systems where it can remain stored over long periods of time. The migration functions almost like a planetary circulatory system, transferring material invisibly between ecological layers while helping regulate global climate itself. The Barreleye participates in this process quietly and indirectly. By feeding on migrating organisms while remaining largely within the deeper mesopelagic zone, it becomes part of the chain carrying surface-derived carbon into the dark ocean interior. The fish drifting motionless beneath faint traces of light is connected, through countless hidden interactions, to atmospheric chemistry occurring far above the waves. Across the twilight zone, millions of similar organisms contribute to this enormous unseen mechanism. Lanternfish, crustaceans, gelatinous predators, siphonophores, and drifting zooplankton all help determine how much carbon the ocean absorbs and how efficiently it is transported downward. The mesopelagic world often appears isolated from the surface, yet its inhabitants exert measurable influence over the planet's climate simply by continuing the ancient movements evolution shaped within the deep Pacific dark. Far from the narrow canyons and drifting prey fields inhabited by the Barreleye, other members of its family move through the deep ocean, carrying their own variations of the same ancient problem. Light is scarce, pressure immense, and visibility measured in fragments. Across millions of years, the fish grouped within the family Opisthoproctidae evolved different solutions to perceiving a world that offers almost nothing to see. Macropinna microstoma, the familiar Barreleye of the North Pacific, represents only one branch within this unusual lineage. The family contains approximately 11 recognized species, collectively known as barreleyes and spookfish, scattered through mesopelagic waters across multiple ocean basins. Even the family name carries a strange history. Derived from Greek, Opisthoproctidae translates loosely to rear-facing anus, an anatomical reference attached by taxonomists with little concern for elegance. Deep-sea biology often accumulates names this way, assembled from obscure physical details noticed under laboratory light long before the living animals themselves were properly understood. What unites these fish is not appearance alone, but a shared evolutionary fixation on vision. Again and again, within the family, anatomy bends toward solving the same environmental challenge, extracting meaningful visual information from near total darkness. Yet the solutions differ dramatically between species, as though evolution experimented repeatedly with entirely different branches of optical physics inside the same lineage. Some species resemble simplified versions of the Barreleye itself, possessing tubular eyes fixed permanently upward with no ability to rotate. These fish remain committed to a single direction of sight, scanning the faint water above for silhouettes crossing the dim remains of downwelling light. Their visual systems sacrifice flexibility for concentrated sensitivity, functioning as specialized detectors aimed continuously toward the surface. Other relatives followed a far stranger path. Among the most remarkable is the brown snout spookfish, Dolichopteryx longypes, a deep-sea species whose eyes abandoned conventional lens-based focusing almost entirely in part of the visual system. Instead of relying solely on curved lenses to direct incoming light onto photoreceptors, portions of the eye contain reflective structures composed of layered crystalline plates. These mirrors gather and redirect light internally, using principles strikingly similar to those employed in reflecting telescopes designed by humans centuries later. In practical terms, the fish evolved a biological mirror. The reflective surfaces inside the brown snout spookfish eye function differently from ordinary vertebrate vision. Rather than refracting light through transparent tissue alone, the curved crystalline mirrors focus incoming illumination onto the retina by reflection, increasing sensitivity under extremely dim conditions. Researchers examining the anatomy described the system as the only known example of a mirror serving as the primary optical element in a vertebrate eye. Elsewhere in the animal kingdom, reflective surfaces exist in simpler visual structures, particularly among invertebrates, but vertebrate eyes overwhelmingly depend upon lenses. The spookfish quietly departed from that convention somewhere in the deep evolutionary past. The result is astonishing not only for its rarity, but for what it reveals about the flexibility of natural selection. Within a single family of fish, evolution produced at least two fundamentally different methods for solving the same problem of deep-sea vision. One lineage refined lenses into elongated tubular detectors enclosed within transparent domes. Another assembled reflective optics from crystalline mirrors arranged inside the eye itself. The Barreleye represents the lens solution, gathering faint traces of light through curved refractive structures carefully tuned to the twilight zone. The spookfish represents the mirror solution, redirecting dim illumination through reflective geometry instead. Both systems achieve functional vision under nearly identical environmental conditions, yet they arrive there through entirely different physical principles. This diversity suggests that the mesopelagic ocean does not impose one perfect answer upon life, only severe constraints within which many strange possibilities emerge. The deep sea repeatedly pressures organisms toward sensitivity, precision, and optical efficiency, but evolution remains free to invent unexpected pathways toward those goals. Somewhere beyond the reach of human observation, across cold layers of Pacific and Atlantic darkness. These distant relatives continue drifting through the water with eyes unlike any others on Earth, each carrying its own interpretation of how to see inside a world where light barely survives at all. Evolution rarely invents in complete isolation. Across the history of life, unrelated organisms facing the same environmental pressures often arrive at remarkably similar solutions, even when separated by immense evolutionary distance. In the deep ocean, this pattern becomes especially visible, as though darkness itself narrows the possibilities available to living bodies until different lineages begin independently, discovering the same biological designs again and again. The tubular eye offers one of the clearest examples. Long before the Barreleye appeared in the North Pacific Twilight Zone, other deep-sea fish lineages had already evolved, elongated, upward, pointing visual systems specialized for collecting dim light. Scientists now believe tubular eyes emerged independently in at least 4 separate groups of mesopelagic fish, not inherited from a shared recent ancestor, but produced repeatedly because the physics of vision at depth leaves relatively few effective alternatives. When sunlight fades into scattered traces overhead, a narrow, highly sensitive optical cylinder becomes one of the best available solutions for detecting silhouettes against weak downwelling illumination. The fish themselves may differ enormously in ancestry, anatomy, and behavior, yet the eyes converge toward the same basic geometry. Evolution experiments continuously, but the deep sea rewards only certain outcomes. This pattern extends far beyond fish alone. Octopuses and squid, belonging to the mollusk lineage, diverged from vertebrate ancestors more than 500 million years ago. Their evolutionary paths separated long before complex eyes resembling modern vertebrate vision existed at all. Yet, over immense stretches of time, cephalopods independently evolved camera-style eyes strikingly similar in structure to human eyes, complete with lenses, irises, and retinas, organized around the same optical principles. The resemblance emerged not through shared inheritance, but because both lineages faced the same challenge, gathering focused visual information efficiently from surrounding light. In the giant squid, this convergence reaches extraordinary scale. Possessing the largest eyes of any known living animal, sometimes approaching 27 centimeters in diameter, the squid carries visual organs roughly comparable in size to a soccer ball. Such immense eyes evolved not for beauty or excess, but for a specific ecological purpose. Deep within the ocean, sperm whales, hunting squid, disturb clouds of bioluminescent organisms as they move through the darkness. The giant squid's enormous eyes appear optimized to detect these faint flashes from great distances, allowing the animal to sense approaching predators before direct contact occurs. Even transparency, one of the Barreleye's defining traits, appears repeatedly across unrelated mesopelagic organisms. Transparent shrimp drift through the twilight zone, almost invisible except for their internal organs. Transparent larvae descend through the water column like living glass. Jellyfish dissolve optically into the surrounding ocean until only drifting movements reveal their presence. None of these organisms share close ancestry with the Barreleye, yet all independently arrived at the same physical strategy. In a habitat where visibility invites predation, becoming transparent provides immediate advantage. Biologists refer to this recurring pattern as convergent evolution, the tendency of separate evolutionary lineages to develop similar traits when exposed to similar environmental constraints. Nowhere does convergence appear more dramatic than in the deep sea because the environment itself imposes such severe limitations. Light is scarce, pressure is immense, food arrives unpredictably. Under these conditions, radically different organisms are funneled repeatedly toward overlapping solutions: elongated eyes, transparency, bioluminescence, hovering locomotion, and hypersensitive sensory systems. The Barreleye belongs fully within this broader pattern while remaining uniquely strange even among its convergent relatives. None of its major features exist entirely alone in nature. Tubular eyes evolved elsewhere. Transparent tissues evolved elsewhere. Hovering ambush predators evolved elsewhere. Yet the precise combination of all these adaptations inside a single vertebrate animal appears nowhere else on Earth. The dome, the rotating green eyes, the suspended posture in cold mesopelagic water, each element reflects pressures shared across the deep ocean, but assembled together, they form something singular. Evolution did not invent the Barreleye from nothing. It gathered familiar solutions from the surrounding physics of darkness and recombined them into one highly specific organism, shaped perfectly for a narrow layer of ocean, where the same environmental demands have been quietly sculpting life for millions upon millions of years. Human beings have spent more than a century trying to study the Twilight Zone, yet almost every confirmed detail about life there has arrived slowly, fragment by fragment, carried upward through conditions that actively erase evidence. The difficulty is not simply that the deep ocean is dark or cold. Those obstacles can be answered with lights, insulated equipment, and powerful vessels. The deeper problem is pressure, the immense physical force pressing inward from every direction, shaping both the animals that live there and the limitations of the tools sent to observe them. At roughly 800 meters below the surface, the surrounding water exerts approximately 80 atmospheres of pressure, nearly 80 times greater than the atmospheric pressure experienced at sea level. Every square centimeter of exposed material must endure a constant compressive force unlike anything present in ordinary surface environments. Human bodies cannot survive direct exposure to such conditions. Most unprotected instruments cannot either. Deep sea research, therefore, begins not with biology, but with engineering. Cameras descending into mesopelagic water must be enclosed inside thick pressure-resistant housings capable of surviving immense compression without warping or flooding. Electrical systems require seals strong enough to prevent microscopic leaks. Sampling containers intended to preserve living organisms must maintain internal pressure during retrieval or risk destroying fragile tissues before scientists can even examine them. Every descent into the deep ocean becomes an experiment, not only in observation, but in preventing the environment itself from crushing the act of observation apart. Even modern remotely operated vehicles only partially solve the problem. Their cameras provide access to living animals inside their native habitat, yet the very presence of the machines alters the scenes being recorded. Bright floodlights cut through an environment where many organisms evolved under permanent dimness. Electric motors generate vibration and noise that travel efficiently through seawater. Propellers disturb the surrounding currents, shifting suspended particles and changing the movement of delicate animals nearby. Researchers watching footage from the deep sea must constantly account for the possibility that the behavior they observe has already been influenced by the arrival of the vehicle itself. Some deep-sea species flee immediately from artificial light. Others freeze motionless. Certain gelatinous organisms fragment or drift unnaturally in altered water flow. The ocean visible through a research camera is never entirely untouched. Pressure-preserving collection systems exist, and can maintain deep-sea organisms inside sealed containers during ascent, allowing tissues to remain closer to their natural condition once brought aboard a research vessel. Yet these systems remain expensive, technically demanding, and comparatively rare. Most mesopelagic sampling still relies on methods not fundamentally different from those used during the early 20th century. Nets lifted slowly upward through changing pressure until whatever survives reaches the surface. As a result, many collected specimens arrive damaged before examination even begins. Soft tissues rupture, gelatinous structures collapse, organs deform under decompression. The Barreleye's transparent dome remains perhaps the clearest symbol of this entire problem. Perfectly stable within its native environment, the structure disintegrates rapidly during ascent, vanishing almost completely by the time researchers handle the specimen directly. For decades, scientists unknowingly studied a distorted version of the fish because the most extraordinary feature could not survive transportation into the surface world. The dome behaves almost like self-destroying evidence, existing fully only under conditions humans struggle to access safely. Its collapse revealed something broader about deep-sea biology itself. Many mesopelagic organisms may possess structures, behaviors, or sensory systems altered or erased entirely by the process used to study them. This realization continues reshaping marine science. Researchers increasingly suspect that the Twilight Zone contains far more biological diversity than current records suggest. For every described species drifting through the mesopelagic dark, several more may remain entirely unknown, either too fragile to survive collection or too rarely encountered for formal identification. Entire evolutionary lineages could exist, suspended between surface and abyss, without ever reaching scientific recognition. The deep ocean, therefore, remains partially hidden not because humanity lacks curiosity, but because the environment itself resists translation into the surface world. Pressure preserves one reality below and destroys it above, leaving researchers to reconstruct fragments of life from evidence altered by the journey into light. For much of modern marine science, the deep ocean arrived at the surface, damaged beyond recognition. Nets scraped blindly through the mesopelagic zone, collecting whatever failed to escape and whatever survived the ascent. Delicate tissues tore apart against mesh. Gelatinous animals disintegrated into translucent residue. Pressure-sensitive organs collapsed before researchers could document them. Entire ecosystems were interpreted through fragments, dragged upward from darkness, flattened into approximations by the violence of collection itself. This method shaped nearly all early understanding of deep-sea life. Scientists inferred behavior from dead anatomy. They reconstructed ecological relationships from partial remains scattered across sorting tables aboard research vessels. The ocean beneath the Twilight Zone became known through interruption rather than observation, through bodies removed from context instead of organisms witnessed alive within their own environment. The arrival of remotely operated vehicles changed that relationship permanently. During the 1980s and 1990s, the first deep-sea ROV systems, capable of prolonged operation within mesopelagic and abyssal depths, began transforming oceanographic research. Unlike earlier submersibles, limited by short dive times and small viewing ports, these tethered, robotic vehicles could remain underwater for extended periods while transmitting live video directly to scientists aboard surface ships. Equipped with lights, cameras, manipulator arms, and navigation systems, they allowed researchers to move slowly through the water column without immediately destroying what they came to study. Among the most influential systems was Embury's ROV Tiburon, the vehicle responsible for recording the living Barreleye in 2004. Tiburon could descend to depths approaching 4,000 meters while carrying high-definition imaging equipment capable of capturing details impossible to observe through older technologies. For the first time, researchers could watch mesopelagic organisms behaving naturally inside the dark environments that shaped them. The impact on marine biology has often been compared to the invention of the microscope. Entire categories of anatomy, behavior, and interaction invisible to trawl net science suddenly emerged into view. The deep ocean ceased being merely a source of damaged specimens and became an observable living world. Some of the discoveries overturned assumptions that had persisted for decades. Siphonophores, the drifting colonial organisms hanging through the water column like living chains, were found reaching extraordinary lengths exceeding 40 or even 50 meters in some species, longer than a blue whale. Trawl nets had missed the true scale of these animals almost entirely because their fragile bodies shattered instantly upon contact. What arrived aboard ships were disconnected fragments impossible to reconstruct accurately. Only sustained ROV observation revealed their immense intact forms suspended through open water. Midwater fish behavior also changed under direct observation. Many mesopelagic species previously assumed to live solitary lives were filmed forming schools and coordinated groups at depth, patterns almost completely absent from net-based evidence. The assumption of isolation had emerged partly because nets separated organisms during capture, erasing whatever spatial relationships existed naturally below. The Barreleye itself became one of the clearest demonstrations of this technological shift. Without the ROV, its transparent dome and rotating eyes would likely have remained unknown indefinitely. The fish had spent decades appearing fundamentally different in death than in life. Observation at depth restored the missing anatomy and corrected the scientific record in a single encounter. Comb jellies, or ctenophores, revealed another hidden reality. These gelatinous predators appeared relatively uncommon in trawl data because their bodies dissolved so easily during collection. Yet ROV surveys showed them drifting through the mesopelagic zone in astonishing abundance. Researchers now suspect they may rank among the most numerous gelatinous predators in the ocean, a conclusion impossible to reach through traditional sampling alone. Gradually, a deeper realization emerged from all these discoveries. The remotely operated vehicle did not merely improve the clarity of ocean imagery. It exposed how incomplete earlier interpretations had been. The problem was never simply that scientists lacked sharper pictures. The problem was that the process used to obtain those pictures altered the organisms before they could be understood. The deep ocean viewed through a net and the deep ocean viewed alive through an ROV are not quite the same place. One contains remains. The other contains behavior, movement, transparency, fragility, and relationships unfolding continuously in darkness. The camera descending quietly into the mesopelagic zone revealed not only new animals, but the limits of the methods that came before it. Within the mesopelagic zone, the Barreleye occupies a narrow but important position inside a much larger web of exchange. It is neither an apex predator nor a passive drifter carried entirely by surrounding currents. Instead, it exists midway through the movement of energy descending from the bright ocean above into the colder layers below, feeding on smaller organisms while remaining vulnerable to larger hunters moving through the same darkness. Its prey consists primarily of zooplankton and gelatinous animals suspended through the water column, tiny crustaceans, drifting siphonophore structures, larval fish, and delicate jelly-like organisms become concentrated packets of surface-derived energy carried downward through migration and marine snow. The Barreleye intercepts this material quietly, converting scattered drifting life into biomass that can then be consumed by larger mesopelagic predators. In this way, the fish functions as an intermediary, a transfer point inside a chain extending from microscopic phytoplankton near the surface to predatory animals deeper in the ocean interior. The relationship becomes even more intricate through its interactions with siphonophores. These colonial predators suspend long feeding structures through the twilight zone, collecting crustaceans and other drifting organisms from the surrounding water. When the Barreleye steals prey directly from siphonophore tentacles, it engages in kleptoparasitism, diverting captured energy away from one predator and into another. The prey itself may have originated near the surface as phytoplankton, consuming sunlight and dissolved carbon dioxide before passing through multiple intermediate organisms on its descent into darkness. Inside the mesopelagic ecosystem, such exchanges occur continuously and silently. Energy rarely travels in straight lines. It is intercepted, redirected, stolen, consumed, and redistributed through thousands of overlapping interactions between organisms drifting through different layers of water. Unlike many migratory mesopelagic fish, the Barreleye appears comparatively stationary in its ecological role. It does not regularly connect distant depth zones through large vertical migrations. Instead, it remains associated with a relatively stable layer of the water column, functioning as a long-term resident rather than a daily commuter between surface and deep environments. This permanence gives the fish a different ecological influence from species constantly moving between layers. A resident predator applies continuous pressure upon the same prey communities over extended periods. By feeding selectively on copepods and small drifting organisms within a narrow depth range, the Barreleye may influence which species persist there and in what relative abundance. Predation does not simply remove individuals. Over evolutionary timescales, it shapes behavior, body structure, migration timing, transparency, reproductive strategies, and even sensory systems among prey populations. The Barreleye participates in this process quietly, helping structure mesopelagic communities through repeated interactions too subtle for direct observation aboard research vessels. Yet, despite this ecological importance, basic population information remains almost entirely absent. No reliable estimate exists for how many Barreleyes inhabit the North Pacific Ocean. Researchers do not know whether the species is comparatively abundant within suitable habitat or exceptionally rare. Its total biomass contribution to the mesopelagic ecosystem remains unmeasured. Even long-term population trends are unknown because sightings remain infrequent and concentrated around limited observation sites such as Monterey Bay. This uncertainty is not unusual within deep-sea ecology. For the vast majority of mesopelagic species, scientists possess only fragments of understanding. They may know the organism's anatomy, approximate diet, or geographic range, while remaining unable to answer more fundamental questions regarding abundance, lifespan, reproductive rates, or ecological resilience. Entire food webs operate beneath the surface, with population structures still largely invisible to science. The difficulty lies partly in scale. The mesopelagic zone represents the largest living habitat on Earth by volume, extending across immense regions of open ocean where direct observation remains sparse and expensive. Small predators like the Barreleye drift through this enormous environment far beyond the reach of continuous monitoring. As a result, Marine biologists increasingly recognize that knowing a species exists is not the same as understanding its ecological role fully. The Barreleye may influence the structure of mesopelagic communities in significant ways while remaining quantitatively almost absent from scientific records. Somewhere between invisibility and importance, the fish continues occupying its precise position within the water column, participating in exchanges of energy and predation that have persisted for millions of years beneath the surface of the North Pacific. Across the surface of the North Pacific, storms, currents, and shifting temperatures shape one of the largest continuous environments on Earth. Beneath those moving waters, far below shipping routes and coastal weather systems, the Barreleye drifts through a specific region of ocean that has quietly sustained its lineage for millions of years. Macropinna microstoma has never been recorded outside the North Pacific Basin. Its range stretches from the waters off California and Baja, Mexico, westward across the open ocean toward Japan and the Kuril Islands. Inhabiting a vast arc of cold mesopelagic habitat suspended beneath one of the planet's largest marine systems. The North Pacific itself covers approximately 165 million square kilometers, making it the largest ocean basin on Earth. From above, its scale appears almost abstract, an immense expanse connecting continental margins separated by thousands of kilometers of open water. Yet within this enormous basin, currents and temperature structures create ecological regions as distinct as forests, deserts, or mountain ranges on land. Along the western coast of North America, one of the most important of these systems flows steadily southward. The California Current carries cold, nutrient-rich water from northern latitudes down the Pacific coast, supporting highly productive surface ecosystems filled with plankton, fish, seabirds, and marine mammals. Nutrients rising into sunlit waters fuel intense phytoplankton growth, and from this productivity begins the long descent of marine snow into the twilight zone below. Everything drifting downward through the mesopelagic ecosystem ultimately traces back to these surface processes. Tiny fragments of dead plankton, fecal pellets, larval remains, and organic debris sink gradually into deeper layers where organisms like copepods, siphonophores, and the Barreleye intercept them. The fish itself remains hidden hundreds of meters below, yet its survival depends intimately upon biological production occurring far above the reach of its vision. Within this broad Pacific system, Monterey Bay became one of the most important windows into the Barreleye's world. Along the coast of central California, the seafloor drops suddenly into Monterey Canyon, one of the deepest submarine canyons bordering North America. The canyon descends beyond 3,000 meters, only a relatively short distance offshore, allowing researchers to access deep mesopelagic environments without traveling far into the open ocean. This unusual geography made the region ideal for sustained deep-sea observation. Research vessels could launch remotely operated vehicles directly above profound depths while remaining close to shore-based institutions and logistical support. The same underwater landscape that shaped nutrient flows and concentrated marine life also enabled repeated scientific encounters with fragile mesopelagic species. It was here, within the dark water surrounding Monterey Canyon, that MBARI researchers captured the now famous footage, revealing the Barreleye's transparent dome and rotating eyes alive for the first time. Other members of the Opisthoproctidae family appear across all major ocean basins, demonstrating that the broader Barreleye body plan succeeds under mesopelagic conditions globally. Tubular eyes, hovering locomotion, and specialized deep-sea vision evolved repeatedly across multiple regions of the world ocean. Yet Macropinna microstoma itself remains confined specifically to the North Pacific. The reasons for this restricted distribution are still uncertain. Temperature likely plays an important role. Mesopelagic ecosystems vary subtly between ocean basins in ways difficult to observe directly, with differences in nutrient cycling, dissolved oxygen, current structure, and prey availability shaping which species persist successfully. The California current and related Pacific systems may create particularly favorable conditions for the barreleye's feeding strategy and sensory adaptations. Current patterns may also influence larval dispersal, preventing populations from establishing permanently outside the North Pacific, despite the apparent suitability of similar depths elsewhere. Even slight changes in temperature or prey composition across geological timescales could isolate populations long enough for specialization to deepen. For now, the Barreleye remains tied to this enormous cold basin spanning the northern Pacific world. Somewhere beneath the shifting surface waters between California and Japan, among drifting crustaceans and descending marine snow, the species continues occupying the same hidden layer of ocean where it evolved, suspended within a geography vast enough that most of its range has likely never been directly observed by human eyes at all. Far above the mesopelagic zone, sunlight touches the surface ocean and disappears into billions of microscopic cells drifting through illuminated water. These phytoplankton, almost invisible individually, absorb carbon dioxide from the atmosphere through photosynthesis, converting dissolved carbon into living tissue that becomes the foundation of marine food webs across the planet. When those organisms die, or when they are consumed by larger animals that later produce waste, fragments begin sinking slowly downward into the dark ocean below. The descent appears gentle, almost insignificant at the scale of individual particles. Tiny remains drift through the water column like underwater snowfall, carrying carbon absorbed from the atmosphere toward deeper layers of the sea. Yet, collectively, this process shapes one of the most important climate mechanisms on Earth. Scientists call it the biological carbon pump, a planetary system through which the ocean removes carbon dioxide from surface circulation and stores it far beneath the waves. The mesopelagic zone, home of the Barreleye, occupies the center of this movement. For many years, researchers imagined marine snow sinking passively toward the abyss, with relatively little interference. More recent work has revealed something far more dynamic. Mesopelagic animals intercept enormous quantities of descending organic matter, consuming, transforming, and redistributing it continuously as it moves through the twilight zone. Small fish, crustaceans, gelatinous predators, siphonophores, and countless drifting organisms all participate in accelerating the transfer of carbon into deeper ocean layers. When mesopelagic animals feed, they convert soft, suspended material into denser fecal pellets and biological debris that sink more rapidly than loose marine snow alone. Migrating organisms also carry carbon physically within their bodies as they travel between surface and deep waters. Predation, digestion, respiration, and decomposition become part of a vast biological engine, moving atmospheric carbon downward through the ocean interior. The scale of this hidden system remained underestimated for decades, partly because the mesopelagic zone itself remained poorly measured. Then, during the early 21st century, improved acoustic surveys and deep-sea observations suggested that global mesopelagic fish biomass might be dramatically larger than previously believed. A study published in 2014 proposed that the Twilight Zone could contain between 1 and 10 billion tons of fish biomass, potentially exceeding earlier estimates by an order of magnitude. If those numbers are even partially correct, the mesopelagic zone may represent the largest reservoir of vertebrate life on Earth by mass. Such a realization changes more than marine biology. It alters climate science itself. Global carbon cycle models attempt to calculate how carbon moves between atmosphere, land, and ocean systems over time. For decades, many of these models treated mesopelagic ecosystems in simplified ways because the organisms inhabiting them remained difficult to observe directly. Yet, if billions of tons of fish and other animals are actively processing carbon within the Twilight Zone every day, then their collective metabolism becomes a major planetary variable rather than a minor ecological detail. Some climate researchers now suspect that existing models may systematically underestimate the ocean's capacity to absorb and transport carbon because they fail to account fully for mesopelagic fauna and their biological activity. The deep ocean is not merely a passive storage reservoir. It is inhabited by immense living communities, continuously modifying how carbon moves through marine systems. Within this context, the Barreleye acquires an unexpected significance. The small fish hovering motionless hundreds of meters beneath the Pacific surface participates directly in these planetary exchanges simply by feeding, digesting, and surviving within its ecological niche. Every drifting copepod consumed, every fragment of marine snow intercepted through the food web contributes in some small, measurable way to the movement of carbon through the ocean interior. The Barreleye itself remains only one tiny data point within an immense, unresolved equation. Researchers still do not know how many exist, how much biomass they collectively represent, or precisely how their feeding behavior influences carbon transport through mesopelagic ecosystems. Yet the broader principle has become increasingly clear. The Twilight Zone is not biologically isolated from Earth's atmosphere. It is connected intimately to it through millions of daily interactions unfolding in darkness. Somewhere around 800 meters below the North Pacific surface, the Barreleye continues drifting through cold water, while above it winds move across continents and carbon circulates through the atmosphere. The connection between those worlds once seemed impossibly distant. Now it appears physical, measurable, and ongoing, carried quietly through the hidden metabolism of the deep ocean itself. Even after the cameras arrived, after the transparent dome was finally seen intact and the rotating eyes observed alive within the dark water, the Barreleye remains surrounded by uncertainty. The footage from Monterey Bay corrected one profound misunderstanding, yet beyond that revelation, the species still drifts through science as something only partially illuminated. Researchers now know more clearly what the fish looks like and roughly how it behaves, but much of its existence remains unwitnessed, hidden inside a habitat too large, too deep, and too difficult to study continuously. Its reproduction is almost entirely unknown. No confirmed Barreleye eggs have ever been identified in the wild. No larval stage has been definitively connected to the adult form. Somewhere within the North Pacific, adult Barreleyes reproduce successfully enough to maintain populations across immense stretches of ocean, yet the beginning of their life cycle has never been directly documented. Marine biologists occasionally encounter transparent fish larvae drifting through mesopelagic waters whose identities remain uncertain. But linking those forms to specific deep-sea adults is extraordinarily difficult. Many species undergo dramatic anatomical changes between larval and mature stages, and without genetic confirmation or direct developmental observation, identification remains speculative. Its lifespan also remains unresolved. Surface fish are often aged through growth rings in bones or ear structures called otoliths, somewhat analogous to tree rings recording cycles of development over time. In the Barreleye, no reliable aging method has yet been applied successfully to living populations. Researchers do not know whether the species survives only a few years or persists through much longer spans in the cold, stable darkness of the mesopelagic zone. Population size remains equally uncertain. No regional estimate exists anywhere across its range. The fish may be uncommon but broadly distributed, or locally abundant yet rarely observed because of its habitat and behavior. Both possibilities remain plausible. Encounters through remotely operated vehicles are infrequent enough that scientists still cannot describe confidently how dense or sparse populations may be in different parts of the North Pacific. Even the fish's relationship with light remains incomplete. The Barreleye evolved extraordinary adaptations for detecting bioluminescence around it, yet researchers still do not know whether the species produces any biological light of its own. No confirmed bioluminescent organs have been identified, but absence of evidence in the deep sea rarely guarantees absence entirely. Countless mesopelagic organisms carry subtle photophores or intermittent light-producing structures difficult to observe under artificial conditions. Its diet, though partially understood through stomach contents and direct observations, remains only fragmentary. Very few specimens have ever been examined in detail, and those represent isolated moments rather than a complete ecological picture. Seasonal variation, geographic differences, and changes across life stages all remain largely undocumented. Even the dome itself may contain functions beyond those already described. Some researchers have speculated that the transparent shield could assist in sensing pressure changes or subtle water vibrations surrounding the fish, perhaps acting as part of a broader sensory system rather than serving only optical protection and light collection. At present, no direct evidence confirms this possibility. The idea remains suspended between hypothesis and observation, another unanswered detail drifting through the literature. Communication presents another unresolved silence. Scientists do not know whether barreleyes interact socially at all. No mating behavior has been observed. No schooling patterns have been confirmed. Whether individuals recognize one another chemically, visually, mechanically, or not at all remains unknown. In many deep-sea environments, encounters between members of the same species may be infrequent enough that communication itself evolves differently from surface ecosystems shaped by constant visual contact. What becomes increasingly clear is that the gap between knowledge and uncertainty is not closing rapidly. Mesopelagic research remains expensive, technically difficult, and dependent upon limited observation opportunities. A single deep-sea expedition can consume years of funding and planning while producing only brief encounters with fragile organisms drifting through vast water columns. The Barreleye therefore continues existing at the edge of scientific understanding, known enough to reveal extraordinary adaptations, unknown enough that basic facts about its life remain unwritten. The transparent dome may now be visible, but much of the animal's existence still moves quietly beyond the reach of sustained human observation, suspended in cold Pacific darkness where answers arrive slowly, if they arrive at all. Long after the first maps of continents were completed, after satellites traced mountain ranges and deserts from orbit with extraordinary precision, the greater part of Earth's ocean still remains only partially seen. As of 2024, Less than 1/5 of the seafloor has been mapped at high resolution, and even that figure describes only the solid ground beneath the water. The immense volume suspended above it, the drifting layers where mesopelagic life moves through darkness between surface and abyss, remains vastly less understood. In many parts of the Pacific, scientists possess clearer images of distant planetary terrain than of the living water columns lying beneath familiar shipping routes. Only a small number of institutions possess the equipment capable of sustained exploration in these environments. Monterey Bay Aquarium Research Institute, NOAA Ocean Exploration, Schmidt Ocean Institute, and several related programs operate much of the world's advanced deep-sea, remotely operated vehicle infrastructure. Their vessels travel across oceans carrying robotic systems descended slowly through darkness on fiber optic cables, transmitting images upward from depths where sunlight vanished hundreds of meters earlier. Each expedition represents years of engineering, funding, and logistical coordination condensed into brief windows of observation beneath the surface. The cost of this work remains immense. A single remotely operated vehicle dive can require tens of thousands of dollars in ship time, fuel, crew support, maintenance, and specialized personnel. A full research expedition may consume millions before the vessel even leaves harbor. Storms interrupt schedules, equipment fails under pressure, cameras malfunction in cold salt water. Entire deployments can be lost to mechanical breakdown, hundreds or thousands of meters below the surface, where retrieval becomes impossible. Yet despite these difficulties, the pace of discovery has begun accelerating again. Autonomous underwater vehicles, known as AUVs, are gradually changing the economics of ocean exploration. Unlike tethered, remotely operated vehicles, these systems can travel independently without remaining physically connected to a ship above. Freed from the drag and limitations of long cables, they can survey broader regions more efficiently, mapping terrain, recording environmental conditions, and scanning layers of the water column for biological activity. Some move silently through darkness for hours or days before returning with stored data gathered across distances once impractical to cover. At the same time, another method has emerged from molecular biology rather than engineering. Environmental DNA sampling, often shortened to eDNA, allows researchers to detect the genetic traces organisms leave suspended in seawater. Skin cells, mucus, waste particles, fragments of tissue all drift invisibly through the ocean carrying identifiable sequences of DNA. By collecting water samples from specific depths and analyzing their genetic contents, scientists can infer which species recently occupied that layer of water, even when no direct visual observation occurs. A fish may remain unseen, yet its biological signature lingers briefly within the surrounding sea, like a fading echo. Acoustic technology has also become increasingly important. Many mesopelagic fish possess swim bladders that reflect sonar pulses strongly enough to reveal vast moving concentrations beneath research vessels. Scientists can now identify enormous biological layers suspended in the twilight zone through sound alone, dense aggregations rising and descending through the darkness during diel vertical migration. Acoustic systems cannot yet identify individual species with precision, but they reveal scales of biomass previously underestimated for decades. Together, these technologies are reshaping deep-sea biology in much the same way the remotely operated vehicle once did. Each new method reveals that earlier assumptions were often incomplete, distorted by limited tools and fragmentary observations. The Barreleye itself became one of the clearest symbols of this pattern, an animal misunderstood for nearly 70 years because the environment that created it also concealed it from accurate examination. The coming decade will likely produce further revisions just as profound. Entire ecological relationships may still remain invisible within the mesopelagic zone, awaiting instruments capable of observing them without disruption. Even as funding remains limited and expeditions rare, the boundary between the known and unknown continues shifting slowly downward through the cold Pacific water, where unseen organisms drift beyond the reach of ordinary light. Far beneath the drifting brightness of the surface ocean, the Barreleye moves through a crowded world that almost never appears in human imagination. The Twilight Zone is not empty water. It is a layered community of hunters, scavengers, drifting colonies, and translucent bodies shaped by pressure, darkness, and the faint residue of light descending from far above. Nearly every animal here carries some anatomical compromise or optical illusion that would appear impossible in shallower seas. Yet, within this environment, these forms become practical, efficient, and strangely elegant. Among the most common neighbors of the Barreleye are the hatchetfish, members of the family Sternoptichidae. Their bodies are compressed into thin metallic plates, narrow enough to nearly vanish when viewed from the side. From below, however, another adaptation becomes visible. Rows of tiny photophores line the underside of the body, emitting controlled bioluminescent light downward into the darkness. This process, known as counterillumination, allows the fish to erase its own silhouette against the dim glow filtering from the surface. Predators looking upward from deeper water see only faint illumination instead of the dark outline of prey. Entire schools drift through the mesopelagic zone, carrying these artificial stars beneath their bodies, matching the brightness above with remarkable precision. Moving among them are the dragonfish, elongated predators belonging to the family Stomiidae. Their bodies are dark enough to absorb nearly all available light, interrupted only by scattered photophores glowing blue or green along the flanks. Many possess enormous teeth too large to fit neatly inside the mouth, curving outward like polished needles. Some species carry an adaptation almost unknown elsewhere in deep-sea vertebrates, the ability to produce red bioluminescent light. Most mesopelagic animals cannot perceive red wavelengths at all, having evolved only for blue-dominated darkness. To a dragonfish, however, this hidden illumination functions like a private searchlight, sweeping invisibly across the water column while nearby prey remains unaware it has been seen. Elsewhere, within the drifting layers, moves the giant amphipod Phronema sedentaria, a crustacean whose behavior appears almost mechanical in its precision. Rather than constructing shelter from external materials, it hollows out the gelatinous body of a salp, consuming the internal tissue while preserving the transparent outer barrel. The amphipod then lives inside this floating cylinder, steering it through the water while carrying eggs protected within the chamber. The structure drifts silently through darkness, like a living submarine assembled from the remains of another organism. Long curtains of siphonophores extend through the mesopelagic as well, colonial animals composed not of one body, but many specialized individuals functioning together as a single organism. Some exceed 40 meters in total length, surpassing even the blue whale as the longest animals ever recorded. Their transparent feeding tentacles trail downward through the water column, collecting crustaceans and zooplankton from the surrounding current. Portions pulse softly with bioluminescent light, while other regions remain nearly invisible, forming drifting structures so delicate that traditional nets often destroy them before they can be studied intact. Not far from these colonies drifts the vampire squid, Vampiroteuthis infernalis, an animal occupying a strange evolutionary position between squid and octopus, yet fully belonging to neither familiar category. Small light-producing organs cover portions of its body, glowing faintly within the darkness. Unlike most cephalopods, however, it abandoned active predation long ago. Rather than hunting living prey, it feeds primarily upon marine snow, collecting drifting organic debris with long filamentous appendages extended patiently into the surrounding water. In a realm shaped by scarce energy, the vampire squid survives not through pursuit, but through quiet accumulation. Together, these animals form an ecological network still only partially visible to science. Predator and prey relationships overlap through shifting vertical layers, linked by migrations, bioluminescent signals, drifting carbon, and invisible chemical traces suspended through the sea. Population sizes remain uncertain. Behavioral interactions are rarely observed directly. Entire communities may exist beyond the reach of current instruments, continuing their slow movements through the Pacific darkness, while human understanding follows far behind. For most of human history, the deep ocean existed less as a place than as an absence. Sailors crossed its surface while imagining immeasurable darkness beneath them, a realm inaccessible to ordinary observation and therefore filled with speculation. Long before instruments descended into the abyss, many naturalists assumed the depths must be nearly empty. The pressure seemed too immense, the cold too severe, the darkness too complete for complex life to persist far below the reach of sunlight. In 1843, the British naturalist Edward Forbes proposed what became known as the azoic hypothesis, the belief that animal life could not survive below roughly 600 meters. The theory appeared reasonable within the scientific understanding of the time. Nets lowered into deep water often returned damaged or barren. Few reliable specimens existed. The ocean floor itself remained almost entirely unseen. Beneath a certain depth, Forbes argued, life simply ended. That certainty began dissolving during the voyage of the HMS Challenger between 1872 and 1876. The expedition traveled across the world's oceans conducting dredging operations at unprecedented depths, retrieving animals from more than 5,000 meters below the surface. Strange crustaceans, worms, mollusks, and fish emerged from darkness once considered biologically impossible. The azoic hypothesis collapsed beneath the accumulating evidence, and modern oceanography was born from the realization that the deep sea was not barren, but inhabited. As additional specimens reached museums and laboratories in the late 19th century, the shock deepened. Anglerfish appeared, carrying luminous appendages protruding from their skulls. Viperfish arrived with teeth too large for their mouths to close naturally. Dragonfish emerged from nets with delicate black bodies, threaded by photophores like living circuitry. These animals contradicted nearly every assumption about how vertebrate life should appear or behave. Evolution, isolated beneath pressure and darkness for millions of years, had produced anatomical solutions unlike anything known from land or shallow seas. Yet even then, the deep ocean remained interpreted mostly through dead specimens hauled violently to the surface. Scientists possessed bodies but not behavior, fragments without context. The water column itself continued to conceal its movements. In 1934, William Beebe and Otis Barton descended together inside the bathysphere to a depth of 923 meters. Suspended from a cable beneath a research vessel, the steel sphere carried them into darkness deeper than any humans had previously visited alive. Through small quartz portholes, Beebe observed luminous organisms drifting through the black water, flashes of blue and green light suspended around the bathysphere like moving constellations. His descriptions sounded so extraordinary that some colleagues questioned their accuracy. Beebe described fish that seemed almost unreal: transparent bodies, glowing organs, impossible teeth, movements unlike anything known near the surface. Yet later discoveries repeatedly confirmed that the deep sea was not exaggeration but underestimation. The reality often proves stranger than the earliest reports. Decades later, Jacques Cousteau helped bring underwater exploration into public consciousness through films and television documentaries during the 1950s and 1960s. Millions watched divers move through coral reefs and coastal ecosystems previously hidden from ordinary view, yet most of this work remained confined to comparatively shallow waters reachable by scuba technology. The mesopelagic zone, where the Barreleye hovered unseen, remained beyond public imagination for decades longer. Again and again, the history of deep-sea exploration has followed the same pattern. Assumptions formed from incomplete evidence persist until new technology descends far enough to overturn them. Entire categories of life emerge suddenly from darkness, carrying anatomies that seem improbable only because human observation arrived so late. The Barreleye belongs to this lineage of revelations, another quiet correction delivered by the ocean to a species still learning how little it has truly seen beneath the surface. Deep beneath the modern Pacific, the Barreleye drifts through an environment that feels detached from ordinary history, yet its lineage reaches backward through time, far beyond the existence of human beings. The family Opisthoproctidae, the Barreleyes and spookfish, possesses fossil relatives extending into the Cretaceous period more than 65 million years ago. Long before modern whales entered the oceans, before grasslands spread across continents, before primates appeared at all, ancestors carrying the foundations of this deep water body plan already moved through ancient seas. That span of time includes catastrophe on a planetary scale. The impact event that ended the age of non-avian dinosaurs altered climates across Earth, collapsed ecosystems, darkened skies, and extinguished vast branches of life, both on land and in the oceans. Entire marine communities disappeared, yet through those transitions, the essential architecture of the Opisthoproctid lineage endured. Tubular eyes adapted for dim upward vision remained useful across geological ages because the physical problem itself remained unchanged. Light still filtered weakly downward through the mesopelagic zone. Predators still needed to detect silhouettes above them against the fading brightness of the distant surface. Evolutionary stability over such immense durations often suggests something important about function. Structures that persist through repeated environmental shifts are usually highly effective solutions to recurring biological pressures. The Barreleye body plan may appear strange when viewed from the surface world, but within the physics of the Twilight Zone, it represents refinement rather than eccentricity. The narrow upward gaze of tubular eyes sacrifices broad visual awareness in exchange for extraordinary sensitivity. In darkness, where photons are scarce, concentration becomes more valuable than panoramic sight. The transparent dome itself remains more mysterious from an evolutionary perspective because soft tissues rarely fossilize. Bones endure compression through sediment and stone. Teeth survive for millions of years. Delicate fluid-filled structures almost never persist long enough to leave clear traces in the geological record. Scientists can follow the ancestry of the eye shape through fossil relatives, yet the precise origin of the dome disappears into evolutionary silence. At some unknown point, natural selection favored a transparent shield above the head, and over countless generations, That structure became integrated into the fish's biology so completely that the modern barreleye cannot function properly without it. Nothing in this process required intention or foresight. Evolution does not move toward elegance, simplicity, or visual harmony. It does not select for what appears familiar or beautiful to human perception. It preserves what survives and reproduces successfully within a given environment. The result may appear graceful, awkward, efficient, grotesque, or astonishing, depending entirely on the observer. The barreleye exists because generation after generation of ancestral fish, carrying slightly advantageous variations, endured more successfully than others within the stable darkness of the mesopelagic Pacific. Over tens of millions of years, those incremental pressures accumulated. Muscles capable of rotating the eyes became more precise. Photoreceptors became increasingly sensitive to dim blue light. The dome acquired optical properties closely matching seawater itself. The body flattened into a hovering form that minimized wasted energy. Piece by piece, natural selection assembled an organism so specifically tuned to one narrow environmental band that removing it from that context causes parts of its anatomy to collapse physically within minutes. The resulting animal appears almost alien, not because it violates biological principles, but because it obeys them so completely within conditions unfamiliar to surface life. The rotating tubular eyes, enclosed within a transparent fluid shield, are not an exception to evolution. They are evolution operating continuously across immense spans of time in a stable, demanding environment where only a limited number of physical solutions remain viable. Given enough generations and a persistent problem, life repeatedly discovers ways to endure. Sometimes, those solutions resemble familiar forms seen across forests, rivers, and shallow seas. Sometimes they drift hundreds of meters beneath the Pacific, carrying transparent skulls and upward-staring green eyes through darkness older than human civilization itself. In the immense stillness of the mesopelagic zone, information moves differently than it does near the surface. Waves do not crash overhead. Sunlight does not flicker through shifting currents. Sound travels long distances through dense, cold water, while movement itself becomes rare enough that even slight disturbances carry meaning. The barreleye exists within this restrained environment, using not one extraordinary sense, but an entire sensory system refined for interpreting signals so faint they would disappear unnoticed in shallower seas. Its vision remains the most striking adaptation. Yet the fish does not rely upon sight alone. Running along the length of the body is the lateral line system, a network of sensory cells embedded beneath the skin that allows fish to detect subtle pressure changes and water displacement. In surface waters crowded with turbulence, waves, and rapid motion, this system competes with constant environmental noise. In the Twilight zone, however, the surrounding water often remains remarkably stable. Tiny disruptions stand out clearly against the background stillness. A drifting siphonophore tentacle brushing through the water column may generate a faint pressure wave before the structure itself becomes visible. The slow movement of a nearby crustacean alters surrounding currents in delicate patterns. Even the approach of another fish can disturb the water enough for the lateral line to register its presence. To the barreleye, movement becomes something partially felt as much as seen, a shifting geometry of pressure unfolding through darkness around the body. The olfactory organs contribute another layer of perception. For decades, scientists mistook these dark structures on the front of the head for the fish's eyes, never realizing their actual role because the transparent dome collapsed before specimens reached the surface. These organs function as highly specialized chemical detectors, continuously sampling seawater for dissolved traces drifting through the mesopelagic current. Minute concentrations of biological compounds can indicate prey nearby, the recent passage of another animal, or the proximity of potential danger. In deep water, chemical information often persists longer than visual signals. A small organism moving through darkness may leave behind a drifting molecular trail, detectable long after its body disappears from sight. The Barreleye's environment rewards sensitivity to these gradients because food remains scarce and encounters between animals may occur only infrequently. Detecting opportunity early conserves energy otherwise wasted on unnecessary movement. Sound also shapes the mesopelagic world differently than it does near coastlines or shallow reefs. Fish perceive sound primarily through the inner ear and, in many species, through the swim bladder, acting as a resonating chamber for pressure waves. The barreleye appears to possess a reduced swim bladder or none at all, suggesting that hearing functions through somewhat different mechanisms than in many shallow water fish. At these depths, surface-generated disturbances become heavily dampened by the overlying water column. Wind, rain, and breaking surf fade into distant, low-frequency vibrations. Instead, the acoustic landscape consists largely of biological and geological sounds carried through darkness. Distant movements, shifting currents, and the low mechanical signatures of living organisms travel efficiently through cold, dense water. The mesopelagic zone is not silent, but its sounds are sparse and widely spaced, separated by long intervals of stillness. Taken together, the Barreleye's senses form a system designed not for speed or constant reaction, but for precision. Every sensory pathway appears calibrated toward minimizing wasted effort in an environment where unnecessary motion carries metabolic cost. Vision gathers the faintest available light from above. The lateral line interprets movement through surrounding water. Olfactory organs sample invisible chemical trails drifting through darkness. Hearing monitors distant disturbances across the cold Pacific depths. The resulting animal behaves with remarkable restraint because its environment rewards patience over aggression. Information arrives slowly here, carried through water in fragments of light, pressure, sound, and chemistry. The barreleye survives by interpreting those fragments accurately before acting at all. In the mesopelagic zone, certainty is rare, energy is precious, and every successful movement begins long before the body itself ever shifts position. There is a particular silence that settles over the history of deep-sea exploration when one realizes how recently humanity learned that entire worlds existed beneath the reach of sunlight. For thousands of years, the ocean depths were imagined as absence, a vast submerged emptiness extending downward beyond anchors and nets. Darkness itself encouraged that assumption. Human beings tend to associate visibility with significance, as though unseen places must therefore contain less life, less complexity, less meaning. The barreleye drifts quietly against that idea. Long before ships crossed the Pacific, before mammals walked upright, before human language formed at all, this fish already existed within the mesopelagic darkness, carrying its transparent dome and rotating eyes through cold water hundreds of meters below the surface. Tens of millions of years passed without a single human mind aware of its presence. Entire civilizations rose and disappeared, while the barreleye continued hovering within the same narrow ecological layer, solving the same ancient problems of light, pressure, and survival. Nothing about its anatomy emerged for observation or admiration. The dome did not become transparent to appear extraordinary, under remotely operated vehicle lights. The eyes did not rotate because strangeness held value. Natural selection shaped these structures slowly, without intention, within a world entirely indifferent to whether surface life ever noticed them at all. The fish simply became what its environment required. That realization changes the emotional scale of the animal itself. The Barreleye is not remarkable because it is bizarre by human standards. It is remarkable because it demonstrates how much of Earth's history unfolded independently of human awareness, beyond observation, beyond mythology, beyond language. Entire evolutionary experiments persisted successfully while remaining completely invisible to the species that would eventually call itself intelligent. Even when science finally encountered the Barreleye, understanding arrived imperfectly. For nearly 70 years, researchers examined specimens whose most important anatomical feature had already vanished during ascent from the deep ocean. The collapsed dome transformed the fish into something misleading before it ever reached the laboratory. Scientists described what they could see honestly and carefully, yet the act of retrieval itself altered the evidence. In that sense, the long misunderstanding surrounding the Barreleye's eyes was never simply a scientific mistake. It revealed a deeper limitation within observation itself. Some environments shape life so completely that removing an organism from its original context changes the organism fundamentally before examination even begins. The surface world imposed its own pressures, temperatures, and physics onto a creature evolved for entirely different conditions. Every decompressed specimen resting on a research table carried two truths simultaneously. It revealed the existence of a real animal from the mesopelagic zone, yet it also concealed essential aspects of what that animal actually was while alive. The dome collapsed because the surrounding environment disappeared. The fish reached human hands, already transformed by the journey upward. There is something quietly humbling in that process. Human knowledge often depends upon bringing distant things closer, isolating them, preserving them, translating them into forms accessible to direct study. Yet the Barreleye demonstrates that some realities resist translation cleanly. Certain structures survive only within the conditions that created them. Remove them from those conditions, and part of the truth dissolves before observation can fully occur. This is not only a biological fact. The deep ocean merely makes it visible in physical form. The Barreleye becomes, in this way, less a curiosity than a mirror held toward human understanding itself. It reminds us that the unknown is not always hidden because it is distant or secretive. Sometimes it remains unknown because our methods of seeing alter the thing being seen. Sometimes the attempt to pull a reality into familiar light changes it irreversibly. Far below the Pacific surface, the fish continues existing entirely outside human narratives. It hunts, drifts, senses pressure, waves, and rotates its green eyes upward through darkness whether anyone watches or not. The ocean required a particular solution, and evolution answered with transparent tissue, tubular vision, patience, and stillness. Human astonishment arrived only afterward, late and incomplete, carried downward by fragile machines into a world that had never needed witnesses to exist at all. Somewhere beneath the shipping lanes, beneath drifting plastic fragments and the faint vibration of distant engines, the mesopelagic world continues almost entirely beyond human attention. The fish, siphonophores, amphipods, comb jellies, and gelatinous colonies of the Twilight zone move through cold water as they always have, suspended within a planetary system older than forests, older than birds, older than humanity itself, Yet the deeper science descends into this realm, the more apparent it becomes that the ocean is not merely unexplored, it is profoundly unmet. Current biological estimates suggest that Earth's oceans may contain somewhere between 700,000 and 1 million species. Fewer than 250,000 have been formally described. Even those numbers carry uncertainty because counting unknown life creates a strange mathematical problem. Researchers are forced to estimate the unseen from fragments, from genetic traces drifting through seawater, from brief encounters on remotely operated vehicle cameras, from damaged specimens rising through pressure gradients that alter them before arrival. The Twilight Zone alone likely contains hundreds of species never documented by science. Some may exist in narrow depth bands spanning only a few hundred meters. Others may occupy regions of ocean visited by research vessels only rarely, if at all. Entire lineages may still drift through darkness, without names, without classification, without any place in human understanding. They feed, reproduce, migrate, avoid predators, and die within ecological systems that remain almost entirely undescribed. This pattern has repeated itself with remarkable consistency across decades of deep-sea exploration. Nearly every major remotely operated vehicle expedition into mesopelagic waters encounters organisms that researchers cannot immediately identify. Sometimes these animals prove to be juvenile stages of known species, transformed almost beyond recognition during development. Sometimes they belong to groups so poorly studied that comparison becomes difficult. And sometimes they appear genuinely unfamiliar, carrying anatomical structures or behaviors absent from existing records. The Barreleye occupies an unusual position within that uncertainty. It is one of the fortunate few. It possesses a formal scientific description, preserved specimens, recorded observations, and a Latin name attached to its existence. Macropinna microstoma has entered human language. Most of its neighbors have not. There is something quietly disorienting about recognizing that the deep ocean does not resemble an unfinished map waiting patiently for discovery. The mesopelagic zone is already complete within itself. Predator and prey relationships function continuously. Nutrients circulate downward through marine snow and upward through migration. Carbon moves through bodies, currents, and darkness according to systems refined over immense stretches of geological time. Long before human beings developed instruments capable of descending into the Twilight Zone, this world was already operating with extraordinary complexity and precision. Human ignorance does not interrupt that operation. It only limits our ability to understand the consequences of altering it. And alteration has already begun reaching downward. Deep-sea trawling extends farther into mesopelagic waters as fishing technologies improve and surface stocks decline. Plastic particles now descend through the water column attached to marine snow, entering food webs at depths once thought insulated from surface pollution. Ocean warming slowly shifts temperature gradients that many deep-sea organisms evolved within over millions of stable years. Deoxygenation zones expand in some regions as climate patterns alter circulation and nutrient flow. What makes these changes especially difficult to measure is not merely the scale of the ocean, but the absence of baseline knowledge. Researchers cannot always determine what has changed because in many cases they do not yet know what undisturbed conditions looked like to begin with. Population sizes remain unmeasured. Species interactions remain undocumented. Entire communities remain unidentified. The Barreleye hovers quietly within that uncertainty. One small vertebrate, surrounded by countless forms of life, still moving beyond the edge of description. Its transparent dome and rotating eyes once seemed impossibly strange because human imagination had not yet adjusted to the realities of the deep ocean. Now the fish stands instead as evidence of how incomplete the adjustment still is. The ocean beneath the sunlight is not empty space awaiting introduction. It is an ancient living system that humanity has only just begun to encounter, and even now, only in fragments carried briefly upward from the dark. Far below the weather of the surface ocean, beyond storms, beyond moonlight, beyond the last fading gradients of blue, the Barreleye drifts through a lifetime that unfolds without sunrise. No dawn ever reaches the depths where it hovers. No horizon brightens. No season announces itself through changing daylight. The fish enters existence in darkness, hunts in darkness, and dies in darkness. Yet nothing about its biology suggests absence or deficiency. Every structure of its body carries the quiet precision of an animal completely at home. To surface creatures, darkness often appears as emptiness because human vision depends so heavily on reflected sunlight. Yet the mesopelagic world is not empty at all. It is saturated with information, only translated into forms unfamiliar to terrestrial senses. Bioluminescent flashes ripple through the water like fragments of coded speech. Chemical traces drift in invisible currents, carrying evidence of prey, predators, injury, reproduction, and decay. Pressure waves travel immense distances through the dense stillness of the ocean, announcing movement long before bodies become visible. Marine snow descends continuously from the illuminated waters above, turning the darkness into a slow-moving weather system of organic material. The Barreleye evolved within this environment for millions of years, and its senses reflect that history with extraordinary specificity. The green tubular eyes enclosed inside the transparent dome are not primitive attempts to compensate for darkness. They are instruments exquisitely tuned to interpret it. The faintest silhouette, crossing the distant glow from above, becomes readable. Tiny fluctuations in blue bioluminescent light become meaningful patterns rather than visual noise. Rotating within their fluid shield, the eyes move with measured precision through a world where nearly every photon matters. Human beings often describe such environments using the language of limitation. Words like blind, dark, hidden, and empty appear naturally in surface imagination because terrestrial life evolved beneath an atmosphere flooded with sunlight. Human eyes perform best in daylight, distinguish fine color variation, and orient themselves toward wide landscapes illuminated from above. The visual system of the Barreleye follows an entirely different logic. Its world contains little color, little direct light, and almost no visual contrast except what appears overhead. Evolution shaped its perception accordingly. Neither system is inherently superior. A human being lowered into mesopelagic darkness without technology would become nearly helpless within minutes, unable to navigate, communicate, or survive. The Barreleye, placed beneath tropical sunlight in warm surface water, would fare no better. Each organism represents a solution to a specific environmental problem, refined slowly through natural selection until perception aligns with survival. There is something quietly humbling in recognizing how flexible life becomes when given enough time. The deep ocean did not resist biology, it invited adaptation. Again and again, evolution entered darkness and discovered new ways to interpret it. Transparent bodies emerged to erase shadows. Bioluminescent organs evolved to communicate across black water. Tubular eyes concentrated distant traces of light. Mirror-based retinas appeared in unrelated species. Entire sensory systems shifted away from reliance on vision altogether, favoring chemistry, vibration, and pressure. The Barreleye belongs to that immense lineage of adjustment. Its dome, its rotating eyes, its hovering stillness, and its patient metabolism all speak the same quiet truth. Life does not require sunlight specifically. It requires usable information, reliable energy, and enough stability for adaptation to accumulate across generations. In the Twilight Zone, darkness itself became habitat, not barrier, not punishment, not void, habitat. And within that habitat, the Barreleye moves with the ease of perfect familiarity. The green cylinders inside its transparent head turn upward through cold water, older than human civilization. Chemical traces drift past like faint signatures. Bioluminescent flashes pulse briefly and disappear. Far above, storms cross the Pacific unnoticed. The fish continues hovering beneath them all, reading the darkness fluently, as naturally as surface creatures once learned to read the light. Long before human beings appeared along coastlines, before grasslands spread across continents, before whales returned to the sea, the ancestors of the Barreleye were already suspended in cold Pacific darkness. Geological eras passed above them like distant weather. Continents shifted, ice sheets advanced and retreated. Entire ecosystems rose into dominance and vanished again. Yet in the mesopelagic waters beneath the sunlight, a lineage of deep-sea fish continued drifting through stable pressure and perpetual night with remarkable continuity. The fossil history of the Opisthoproctidae family reaches back beyond 65 million years, extending to a world still occupied by non-avian dinosaurs. Since that distant period, Earth has experienced repeated biological crises. The end Cretaceous extinction erased nearly 3/4 of known species. Later, climatic upheavals altered ocean circulation, reshaped coastlines, and transformed atmospheric chemistry. Surface ecosystems repeatedly fractured under changing conditions, tied closely to sunlight, temperature, and plant productivity. The deep ocean responded differently, not untouched, not invulnerable, but insulated. Most surface ecosystems depend directly or indirectly on photosynthesis occurring beneath sunlight. Forests, grasslands, coral reefs, and terrestrial food webs ultimately trace their energy back to the conversion of solar radiation into biological material. When catastrophic events interrupt that process, collapse spreads rapidly. Ash clouds block light, temperatures shift, plant life declines. Herbivores disappear, predators follow. The Twilight Zone operates according to another rhythm. Energy still originates at the surface, carried downward through marine snow and migrating organisms. Yet the system remains partially separated from the immediate instability of weather and daylight. Darkness does not suddenly arrive there during catastrophe because darkness was already present. Cold does not abruptly dominate because cold already shaped every metabolic process. Pressure remains constant while storms, impacts, and atmospheric disruptions unfold far above. There is something quietly striking in realizing that certain animals endured planetary crises precisely because their world was already disconnected from the systems that failed. The Barreleye persisted not through force or aggression, but through extraordinary specialization. Its biology asks for very little beyond the continued existence of a narrow environmental band within the North Pacific. Cold water, stable pressure, faint downwelling light, suspended prey drifting through darkness. Evolution refined the fish toward those exact conditions until nearly every structure of its body reflected them with precision. Its transparent dome only functions properly at depth. Its rotating tubular eyes interpret dim light almost useless to surface animals. Its slow, hovering posture conserves energy within a habitat where food arrives gradually. Even its small mouth and delicate fins speak the language of efficiency rather than competition. Nothing about the animal suggests dominance in the familiar terrestrial sense. It survives instead through alignment with its environment so complete that the boundary between organism and habitat begins to blur. In evolutionary terms, persistence rarely belongs to the strongest in any absolute sense. It belongs to whatever remains compatible with changing conditions long enough to continue reproducing. A species can possess immense speed, strength, intelligence, or size, and still disappear quickly if the surrounding system shifts beyond what its biology can tolerate. Another species may appear fragile or strange by comparison, and yet persist for tens of millions of years, simply because the environment it depends upon remains relatively stable. The Barreleye embodies that quieter form of endurance. For longer than human beings have existed, it has occupied nearly the same depths beneath the North Pacific, reading faint traces of light through green tubular eyes while continents altered above. Ancient storms passed unnoticed overhead. Meteor impacts darkened skies already irrelevant to its daily existence. Ice ages cooled surface oceans while the deep remained comparatively steady. Generation after generation continued hovering through the twilight zone, suspended inside a world measured less by sudden events than by geological patience. There is no triumph in that persistence, no intention behind it, no lesson deliberately offered. The fish simply remained fitted to its place, while countless other forms disappeared from theirs. And so, the Barreleye continues now, as it did long before human memory, drifting beneath the illuminated surface of Earth, carrying within its transparent dome the evidence that survival is sometimes not about conquering a world, but about belonging completely to one. Far beneath the Pacific surface, beyond the reach of weather forecasts, cargo routes, fishing lights, and human conversation, a small fish remains suspended in cold water nearly 800 meters down. It hovers there almost motionless, balanced within pressure heavy enough to collapse lungs instantly, surrounded by darkness so complete that even the concept of day has almost no meaning. The ocean, at that depth, does not resemble silence exactly. Tiny pressure waves move through the water. Marine snow drifts downward from distant illuminated layers above. Occasional flashes of bioluminescence pulse briefly and vanish again. Within that immense blackness, the Barreleye continues watching. Its green tubular eyes are angled upward through the transparent dome that encloses them, scanning the faintest traces of downwelling light filtering from the world above. The dome is intact there. The delicate fluid-filled shield holds its shape perfectly beneath the surrounding pressure, clear enough for the fish to read silhouettes passing overhead with extraordinary precision. Nothing about the animal appears unfinished or experimental. Every structure belongs exactly where evolution placed it. Human beings know this creature exists now. Cameras have observed it alive. Researchers have recorded its rotating eyes, its hovering posture, its transparent skull. Scientific literature describes its anatomy in careful detail. Yet beyond those fragments, uncertainty still surrounds nearly every aspect of its existence. Its reproduction remains unseen. Its lifespan remains unknown. Its population size has never been measured. Entire portions of its behavior continue unfolding in darkness without observation. The footage captured in the early years of deep-sea, remotely operated vehicles did not reveal mastery over this world. It revealed proximity. A temporary opening appeared between two environments that rarely touch. For a few moments, bright artificial lights crossed into mesopelagic darkness, and illuminated an organism that had evolved entirely outside human awareness. The result felt almost unreal, not because the fish resembled fantasy, but because it exposed how incomplete human understanding of the ocean still remains. The transparent dome itself became part of that lesson. At depth, it exists clearly and naturally, but when brought toward the surface, the structure collapses and dissolves. The evidence destroys itself during retrieval. For decades, scientists misidentified the fish because the defining feature vanished before examination could begin. The Barreleye was never hiding intentionally. Human observation simply altered the thing being observed. There is something quietly important in that fact. Some parts of the natural world cannot be fully separated from the conditions that created them. Remove them from pressure, darkness, temperature, and depth, and essential truths disappear before they can be studied directly. Science continues approaching the deep sea with increasingly sophisticated tools, yet the ocean still imposes limits on what can be carried upward intact. That limitation is not failure, it is scale. The ocean is older, larger, and more continuous than human civilization. It contains systems that operated for hundreds of millions of years before the first research vessel crossed its surface. The Barreleye never required human understanding to persist. Through extinction events, climate shifts, drifting continents, and changing seas, it remained within the Twilight Zone, reading darkness through green rotating eyes while surface worlds transformed overhead. And it remains there now, not as symbol or metaphor, but as living biology, continuing independently in cold Pacific water. Somewhere beneath the illuminated skin of the ocean, a transparent dome still catches faint traces of light. Small fins hold position against almost imperceptible currents. Bioluminescent flashes pulse briefly in surrounding darkness. Marine snow continues descending from distant surface ecosystems above. The fish waits within all of it patiently, perfectly adapted to a world human beings have only barely entered. Rest now. The Pacific remains vast beyond the edges of maps and memory. And somewhere in that immense black water, nearly 800 meters down, the Barreleye is still hovering quietly in the dark, its green eyes turned upward, still watching a world we have only just begun to glimpse.